Good morning, all. Am I audible? Hello. તમારામાં સંભળાય છે? Hello. Hello. Good morning, all. Am I
બોલો મંટુ કુમાર કઈક તો
બોલો
બોલો
સાહેબ.
ક્યાં
બસ
છો
મેં કીધું બધા સાયલેન્ટ છે તો મેં કીધું પછી
અરે ભાઈ મેં તમને અનમ્યુટ જોયા અને હું અનમ્યુટ એટલે અમે વાતો કરીએ તાર
શું છે કેવું છે
પાછળ
સાહેબ ફાર્માસિસ્ટ ડે?
બહુ સારો રહ્યો ને કાલ તો ના મજા આવે
નહીં આપણા આખા ગુજરાત ઇન્ડિયામાં લગભગ ગુજરાતે વધારે સેલિબ્રેટ કર્યું છે આખા ઇન્ડિયામાં.
તો પછી એને પબ્લિશ કરો બરાબર
હા એ બધા રિપોર્ટ્સ બનાવીને પછી પબ્લિશ કરીશું આપણે.
હમ. ક્યાં પ્રોબ્લેમ છે ક્યાં અટક્યું છે અત્યારે
શું છે કેવું છે સાહેબ ફાર્માસિસ્ટ ડે?
બહુ સારો રહ્યો ને કાલ તો ના મજા આવે
નહીં આપણા આખા ગુજરાત ઇન્ડિયામાં લગભગ ગુજરાતે વધારે સેલિબ્રેટ કર્યું છે આખા ઇન્ડિયામાં.
પછી એને પબ્લિશ કરો
હા એ બધા રિપોર્ટ્સ બનાવીને પછી પબ્લિશ કરીશું આપણે. હા હા બોલો બોલો જલ્દી.
ક્યાં પ્રોબ્લેમ છે ક્યાં અટક્યું છે અત્યારે
Sir, actually we are waiting for
બહુ સારો રહ્યો
one
ને કાલ
member.
તો ના મજા આવે
નેક્સ્ટ આપણે ફિલ કરવું પડશે ને ફિલ કર્યા પછી આપણને બધાને પછી એડજસ્ટ કરવું. હા હમ હમ હા
Okay. Good morning, all, and sorry for the inconvenience today. Excellence and a very good morning to all the delegates, ladies and gentlemen. I, Hita Kachya, Assistant Professor and Course Coordinator of today's Refresher course, feeling privileged to extend to you all a very warm welcome on behalf of Induka Kaipkawala College of Pharmacy. Yesterday was the World Pharmacist Day, and we are feeling proud to organize
Excellence and a very good morning to
Gujarat
all
State
the
Pharmacy
delegates,
Council.
ladies and gentlemen. I, Hita Kachya, Assistant.
આ વસ્તુ કહેતી હતી કે આવું સંભળાય છે. Just yesterday was World Pharmacist Day, and we are feeling proud to organize Gujarat State Pharmacy Council sponsored today's online refresher course for registered pharmacists. Prayer should be the key of the day and lock of the night. Let's connect out the mind and heart with prayer.
Was World Pharmacist Day, and we are feeling proud to organize today's online refresher course for registered pharmacists. Prayer should be the key of the day and lock of the night. Let's connect out the mind and heart with prayer. Was World Pharmacist Day, and we are proud to organize today's online refresher.
Hello, good morning to all.
સાથે આવે છે કંઈ?
Hello, good morning to all. Thank you.
Done?
Yep.
Prayer should be the key of the day and lock of the night.
Hello,
Let's
good morning to
connect
all.
out mind and heart with prayer. સાથે આવે છે કંઈ?
Hello, good morning to all.
Now, I would like to request Dr. Harshavi Patel, ma'am, Principal ISCP and Course Director of today's refresher course, to introduce our Chairman of Charutar Vidya Mandal University, Engineer Bhikubai Patel, sir, GSPC representative and chief guest of today's function, Dr. Montakumar Patel, sir. Over to you, madam.
Okay. Good morning to all participants and organizer. First of all, outset, I am thankful to GSPC in the days of offline, this kind of online refresher course organizations, specifically Montu Patel sir and their subordinates. It's my proud and privilege to introduce the eminent personalities who are involved in this. So, outset, I'm thankful to our president, Dr. Engineer Sri Bhikubai Patel sir, president of Charutar University. This year we had recognized at CVM University. He is also chairman of Charutar Vidya Mandal, and it is a very young university. But even though Charutar Vidya Mandal has been established 75 years before, so Bhikubai Patel sir is basically civil engineering or engineer holding. He is having various business, but he has got this leadership natures from his father. He was a sarpanch of Baroda village, which is a nearby Anand. And from 3 years, he is our leader, he is our mentor, and his steps towards the word means CVM to put some status recognitions. So he is encouraging us, supporting us for such kind of events. So I am thankful to him. I am thankful to Charutar Vidya Mandal and Charutar University. Then Sri Montu Patel sir, he is a president of Gujarat State Pharmacy Council since 2015. He is a young dynamic leader. So he is in his blood. It is a warm and hot blood is circulating. So he is always towards doing some activity. Last year he has visited our refresher course second valedictory session, and this year he has come as a inaugural sessions for that. He has basic degree of BPharm and MPharm in pharmacy from Swaminarayan College of Pharmacy. He is at present pursuing his PhD at Saurashtra University. He is involved in this noble, our profession is a noble profession, noble professions, and he is also actively working in GSPC for certain activities like drug information centers, and updating of the portals. So many things has been made digitalizations, and before lockdown, it was just in thought if the refresher course can be done without visiting of the college for the participants. So this has come in actual reality, and it has come in force due to forceful by COVID-19. So I welcome you, sir, on this inaugural session of today's refresher course. Our another guest, due to some problem, he could not make to attend this. Some problem is there. Dr. C.N. Patel, sir, he is a principal and professor of Sarojini College, and he is involved in teaching field since last 30 years. He is active researchers, active leader, and actively acting in academic as well as administrative. Presently, he is a dean of the Gujarat Technological University. He is a member of board of governance of GTU, and he has also remained dean and members of the faculty in North Gujarat HNG University. He is also remains as a life member of certain bodies, APTI, FICS, then alumni associations, and so many other. He means he's a good leader which comes from his NSS activity. He is a recipient of Best NSS Program Officer Award and recipient of Faculty Award. And from the part of research, his students got Best Research Award R.V. Patel. So we welcome you, sir. Even though in spite of your presence, physical is means it is not visible, but you are present with us. Thank you, sir, for with us. So once and the coordinator of this refresher course, Mrs. Heta Kachia and Mrs. Jinal Tandel, their active efforts could make this event possible. So I congratulate both of them. Even though I congratulate the whole team of ISCP who are actively working with for this. So wish you good day ahead for today and tomorrow. Enjoy this lectures and get some gains from this refresher course. Thank you.
Thank you, ma'am. Uh, I would like to present a brief overview about today's refresher course. We are going to organize 6th refresher course by continuous follow-up from registered pharmacist to our institute. I had made discussion with the principal ma'am and our staff members who are working here with respect to how the various issues the registered pharmacists are facing in routine days during dispensing medicines, out to be groomed or trained the registered pharmacist by refresher course. The objective of refresher course is to boost up the self-confidence and morale of the registered pharmacist and to upgrade the technical knowledge of pharmacy professionals, hospital pharmacist, community pharmacist, pharmacist with medicine, business, academicians, and all the registered pharmacists. This refresher course will enrich the participants about roles of pharmacist in responsible use of medicine, and therefore with the objective we would plan today's refresher course for upliftment of budding pharmacist. Concluding my talk, I would add that the online feedback form will be given to you and that you have to duly fill and submit, as well as you have to appear for the online objective test which will be held tomorrow on 27th September at the end of the refresher course. I hope you will have a good time in the lectures and I warmly welcome you again all. Now I would like to invite Chief Guest and GSPC member Dr. Montakumar Patel sir to deliver keynote address. Over to you, sir.
ગુડ મોર્નિંગ ટુ ઓલ અને હેપી ફાર્માસિસ્ટ ડે. ગઈકાલે તો આપણે આજે પ્રોગ્રામ છે એટલે આજે બધા ફાર્માસિસ્ટ મિત્રોને ફાર્માસિસ્ટ ડે ની શુભકામનાઓ. અને ઇન્દુ કાકા ઇફકો વાળા જે ફાર્મસી કોલેજ છે લાસ્ટ યર મેં વિઝિટ પણ કરી હતી. આ વર્ષે આપણે ઓનલાઈન કનેક્ટ કર્યું છે અને જે બે સ્ટાફ ફેકલ્ટી છે ઉસ્માનભાઈ અને બધા સ્ટાફ એમને જે 2 દિવસનો રિફ્રેશર કોર્સ ઓનલાઈન, ઓફલાઈનમાં તો ચલો બધા કરતા અત્યાર સુધી બધા કરતા, પણ ઓનલાઈન કરવો એક બહુ ડિફિકલ્ટ ટાસ્ક છે કે ઓનલાઈન કનેક્ટ કરવું, બધાને સાંભળવું. કારણ કે પેલું તો શું ફેસ ટુ ફેસ હોય તો તમે એકબીજા સાથે વાતચીત કરીને સોર્ટ આઉટ કરી શકો, પણ ઓનલાઈનમાં થોડા ઘણા પ્રોબ્લેમ્સ રહેતા હોય છે અને એમાંથી આપણે આગળ વધીને કેમનું સક્સેસફુલ રિફ્રેશર કોર્સ આપણે કરી શકીએ એના માટે બધાનું કોઓપરેશન જરૂરી હોય છે. ને ડોક્ટર હર્ષાબેન પટેલ પ્રિન્સિપાલ શ્રી અને એમનો સારો પ્રયાસ છે કે એ બાજુના તમારા જે પણ સ્ટુડન્ટ્સ હોય એ લોકોને રેગ્યુલરલી રિફ્રેશર કોર્સ થાય એ માટે ચિંતિત હોય છે. તો એ પણ એક સારું જે પાસ આઉટ સ્ટુડન્ટ માટે એક સારા એ કહેવાય. તો ઇન્દુ કાકા ઇફકો વાળા અને ચારુસત યુનિવર્સિટી જે આ 2 દિવસનું રિફ્રેશર કોર્સનું આયોજન કર્યું છે એના માટે ગુજરાત સ્ટેટ ફાર્મસી કાઉન્સિલ બધી આભાર વ્યક્ત કરું છું. અને કાઉન્સિલના જે પણ પ્રોગ્રામ્સ હોય છે એ આપ સામે ચાલીને દર વખતે સહયોગ લઈને તમે કામ કરો છો એના માટે પણ મારા બધા મેમ્બર વતી આપનો આભાર વ્યક્ત કરું છું. અને 2 દિવસનો રિફ્રેશર કોર્સ બધા ફાર્માસિસ્ટ મિત્રો હાજર હશે એટલે આમ તો ફેસ ટુ ફેસ મળવું શક્ય નથી અત્યારે, પણ ઓનલાઈન આપણે જે કનેક્ટ થવાનો એક મોકો મળ્યો છે રિફ્રેશર કોર્સ દ્વારા, તો એમાં કાઉન્સિલની વાત કરીએ તો આપણે બધું જે કાઉન્સિલની રિન્યુઅલ પ્રોસેસ, જેવી કે તમારે આમ તો બધા એક દિવસનું શું કે કાઉન્સિલ આવવું પડે, કાઉન્સિલ આવે પછી કાં તો કોઈની સાથે મોકલાવવું પડે આ બધા જ પ્રોબ્લેમ જ્યારે અમારી કમ્યુનિટી આવી ત્યારે બધાને ફેસ થયા. તો જે વર્ષો જૂના પ્રોબ્લેમ્સ હતા એ અમારી કમ્યુનિટી આવ્યા પછી ડિજિટલાઇઝેશન તરફ આગળ વધ્યા અને એમાં જે નરેન્દ્ર મોદી સાહેબનું જે સપનું હતું કે ડિજિટલ ઇન્ડિયા એને હેઠળ આપણે કાઉન્સિલનું વેબસાઇટનું ડેવલપમેન્ટ કર્યું અને પછી એનું એન્ડ્રોઈડ એપ અને iOS એપનું પણ ડેવલપમેન્ટ કર્યું. તો આ બધું જ અત્યારે ઓનલાઈન રનિંગ મોડમાં છે અને તમે ઘર જસ્ટ ખાલી 3 સ્ટેપમાં તમારા લાસ્ટ રિન્યુઅલની રિસિપ્ટ અને રિફ્રેશર કોર્સનું સર્ટિફિકેટ આટલું અપલોડ કરો અને પેમેન્ટ કરો 3જા સ્ટેપમાં, એટલે તમારી રિસિપ્ટ ઘરે બેઠા ઘરે આવી જાય. એટલે એકદમ સિમ્પલી પ્રોસેસ આપણે ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલે કરી દીધી છે, જેથી કરીને કોઈ ફાર્માસિસ્ટ મિત્રોનો સમય બગડે નહીં અને પોતાનો એક દિવસની ડ્યુટીમાં જોબ અથવા તો બિઝનેસ કરતા હોય તો રજા ના પાડવી પડે. તો આ બધું જ ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલના મેમ્બરોએ ધ્યાને લઈને આ બધા કામ કરેલા છે. બીજું છે કે જોબ પોર્ટલ. હવે લોકડાઉનમાં અને લોકડાઉન પહેલા બહુ જ બધા શું કહેવાય જોબના ઇસ્યુ વર્ષોથી ચાલતા આવે છે ફાર્માસિસ્ટોના. ભાઈ કોઈ સારા કેન્ડિડેટ હોય પણ કોઈનો રેફરન્સ ના મળતો હોય અથવા તો પ્લેટફોર્મ ના મળતું હોય, કારણ કે તમને ખબર છે તમારી ઘરની બાજુમાં જોબની જરૂર હોય પરંતુ તમને ના ખબર હોય કે આ મેડિકલ સ્ટોર અથવા તો આ ઇન્ડસ્ટ્રીમાં જોબની વેકેન્સી છે. તો એના માટે કાઉન્સિલે એપ્રોચ કર્યો કે ભાઈ આપણે એક આવું પ્લેટફોર્મનું આયોજન કરીએ જેમાં ફાર્માસિસ્ટ પોતાની પ્રોફાઇલ મૂકી શકે, ચાહે ફ્રેશ ફાર્માસિસ્ટ હોય કે એક્સપિરિયન્સ ફાર્માસિસ્ટ હોય. અગર એને જો જોબ બદલવી છે તો આપણે એક પ્રોફાઇલ મૂકવાનું એક પ્લેટફોર્મ બનાવી અને આપણે જોબ પોર્ટલ બનાવ્યું છે અને ગુજરાત સ્ટેટ ફાર્મસી કાઉન્સિલની વેબસાઇટ ઉપર એક્ટિવ છે અને એમાં રિટેલ, હોલસેલ, ક્લિનિકલ, એકેડેમિક બધી જ ટાઈપની જોબ પ્રોફાઇલ અપડેટ થતી હોય છે. ને સામે કંપની વાળાને અમે એપ્રોચ કર્યો, દરેક ઇન્સ્ટિટ્યૂટને એપ્રોચ કર્યો, બધાને મેલ કર્યા કે ભાઈ તમારે આ પ્લેટફોર્મ ઉપર તમારી રિક્રુટમેન્ટ મૂકવી હોય તો તમે મૂકી શકો છો અને ફ્રી ઓફ કોસ્ટ છે, એમાં કોઈ પણ ચાર્જ લેવાનો નથી. કારણ કે આમ બધા જે જોબ પોર્ટલ હોય એક સેલેરી લેતા હોય, 2 સેલેરી લેતા હોય, પરંતુ આપણે ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલે ફાર્માસિસ્ટોને ફ્રી ઓફ કોસ્ટ કે એમના મદદરૂપ એમના કેરિયરમાં થઈ શકે એના માટે એક આ પ્રોગ્રામ જોબ પોર્ટલનું આયોજન કર્યું છે. દર વીકલી આપણે આ જોબ પોર્ટલ ઉપર જેટલી બી નવી રિક્રુટમેન્ટ બહાર આવે છે એની પણ આપણે દરેકને બલ્ક મેલ દ્વારા જાણ પણ કરીએ છીએ. લગભગ બધાને મેલ મળતો જ હશે. જે લોકોના મેલ અપડેટ નહીં હોય એ લોકોને નહીં મળે, જેના અપડેટેડ હશે એ લોકોને મળશે. એટલે કાઉન્સિલનો તમે મેક્સિમમ ઉપયોગ જો તમારી પ્રોફાઇલ અપડેટ રાખશો તો જ કરી શકશો. તો આ બધી જ ઇન્ફોર્મેશન કાઉન્સિલની વેબસાઇટ દ્વારા અને બલ્ક મેલ દ્વારા ફ્રી ઓફ કોસ્ટ આપવામાં આવે છે. બીજો એક પ્રશ્ન હમણાં ઘણા વર્ષોથી તમને એ બી હશે કે ભાઈ આપણે ઘરની બહાર નીકળીએ એટલે 5-10 તો મેડિકલ હોય જ અને 5-10 મેડિકલ સ્ટોરમાં ફાર્માસિસ્ટ કોણ છે એ પણ આપણને ખબર ના હોય, કારણ કે તમે પૂછી બી ના શકો. ઘણા સામાન્ય જનતાને તો ફરક ના પડે, પરંતુ આપણે જેવા ફાર્માસિસ્ટ મિત્રો જો જાગૃત હશે અને જો બહાર નીકળતા 5-10 મેડિકલ સ્ટોર હોય અને તમે પૂછશો કે એમાં ફાર્માસિસ્ટ કોણ છે, ઇવન સામાન્ય જનતા પણ પૂછી શકે છે કે ભાઈ હું જે પણ દવા લઈ રહ્યો છું એ ફાર્માસિસ્ટ થ્રુ જ લઈ રહ્યો છું, પરંતુ આ જાગૃતતાનો અભાવ છે અને આ જાગૃતતા આપણે બધા ભેગા થઈને અપાવવી પડશે. એના માટે કાઉન્સિલે ઘણા એવા ઇન્ડસ્ટ્રીમાં જોબ કરતા હતા જેનો 18 લાખ સેલેરી હતો. ઇવન જાયડસ, ટોરેન્ટ, કેડિલા, ઇન્ટા, સન ફાર્મા બધામાં સારો પેકેજ 50 લાખના પેકેજ વાળા લોકોએ પણ લાયસન્સ રેન્ટ આઉટ કરેલા હતા. તો આપણા પ્રોફેશનનું જે ડિવેલ્યુએશન થઈ રહ્યું છે આપણા લીધે જ થઈ રહ્યું છે. તો આ એક ફાર્માસિસ્ટ ડે નિમિત્તે બધાએ આપણે કરવું જોઈએ કે ભાઈ આપણો જે સર્ટિફિકેટ કાઉન્સિલ આપણને ફાર્માસિસ્ટ તરીકેની જે આઈડેન્ટિટી કાઉન્સિલ પ્રોવાઈડ કરે છે એનું કોમર્શિયલાઇઝેશન ના કરવું જોઈએ, કારણ કે એ વસ્તુ આપણું પ્રોફેશનલી છે. કારણ કે કોઈ પણ ડોક્ટર અથવા તો સીએ, તમે ખાલી સીએ નું એક્ઝામ્પલ લો તો તમે કોઈ પણ એક તમારું ઇન્કમ ટેક્સનું રિટર્ન હોય અથવા તો કંઈ પણ વસ્તુ હોય, 10 વાર તમારા બેંક સ્ટેટમેન્ટ અથવા તો બેંકની જે પણ એન્ટ્રીઝ ચેક કર્યા વગર સાઇન નથી કરતા હોતા, કારણ કે એમને પોતાની સર્ટિફિકેટ સીએને જે મળે છે એની એક વેલ્યુ હશે રજીસ્ટ્રેશન નંબરની. એમ ડોક્ટર હોસ્પિટલ ખોલીને કોઈ બીજાને પ્રેક્ટિસ કરવા નથી આપી દેતો, કારણ કે એને બી એક ખતરો હોય છે કે મારો હોસ્પિટલ બંધ થઈ જશે. એમ જસ્ટ ખાલી આપણા ફાર્માસિસ્ટ મિત્રો જેવા છે કે જે કોઈ સિરિયસલી લેતું નથી, કારણ કે આપણે ફાર્માસિસ્ટનું રજીસ્ટ્રેશન કેટલું ઇમ્પોર્ટન્ટ છે એ અત્યારે નથી સમજાતું. કારણ કે જે લોકોને તકલીફ પડી છે, હમણાં જે રિસન્ટલી રાજકોટમાં એક કેસ થયેલો હતો એમાં ફાર્માસિસ્ટને 6 મહિનાની જેલની સજા પણ થઈ છે. એક ઇન્જેક્શન ખાલી નામ ચેન્જના કારણે એક નાના બાળકનું અવસાન થયું તો ના ડોક્ટરે એની જિમ્મેવારી લીધી, ના નર્સે લીધી, ના કમ્પાઉન્ડરે લીધી જે મેડિકલ સ્ટોરમાં બેસતા હોય એમને, પણ ફાર્માસિસ્ટનું લાયસન્સ વાન રેકોર્ડ હતું એટલે એને જેલની સજા થઈ. એટલે આ એક રિયલ સત્ય છે. અને અત્યારના જમાનામાં જ્યારે આટલી બધી લાખો દવાઓ નવી શોધાવાની હોય, નવા એડવર્સ ડ્રગ રિએક્શન આવે છે, તો ક્યાંય પણ મેડિકલ ઇમરજન્સી અથવા તો ક્યાંય પણ કોઈ પેશન્ટનું તમારા લાયસન્સ ઉપર મોત થયું તો તમે જેલના ભેગું જ જશો, એ 100%ની ખાતરી છે. હમણાં બી તમે જોયું છે કે એક્ટમેરા જે ટોસિલિઝુમેબ ઇન્જેક્શન ડુપ્લિકેટ પકડાયા, એમાં પણ ફાર્માસિસ્ટ ઇન્વોલ્વ હતા. તો એ બધા અત્યારે જેલમાં કેસમાં ચાલુ થઈ ગયા છે અત્યારે એમના ધક્કાને મે બી એમને જેલ પણ થશે. તો આપણું જે રજીસ્ટ્રેશન સર્ટિફિકેટ છે એ આપણે પોતાના માટે પ્રોફેશનલી યુઝ કરવું જોઈએ. કારણ કે તમે બિલ ઉપર તમારી સિગ્નેચર કમ્પલસરી છે. જો તમે એ નહીં કરો તો તમે જ્યારે બી ડ્રગ લાયસન્સ એપ્લાય કરો છો ત્યારે તમારી પાસે એફિડેવિટ લેવામાં આવે છે 50 રૂપિયા કે 100 રૂપિયાના સ્ટેમ્પ પેપર. દવાનું વેચાણ, ખરીદ અને વેચાણ મારી હાજરીમાં જ થશે, અગર નહીં થાય તો હું જ જિમ્મેવાર છું. એ બધું તમે 100ના સ્ટેમ્પ પેપર પર લખીને આપો છો. આનો ઉપયોગ હવે કોને જેટલા લેવલે કરવો હોય એટલા લેવલે કરી શકે છે, પરંતુ કાઉન્સિલ ફાર્માસિસ્ટોના હિત માટે છે, અહિત માટે નથી. અગર જો અમે આના ઉપર લીગલી એક્શન લેવા જઈએ તો તમે પણ જેલ ભેગા થઈ શકો છો. પરંતુ અત્યારે આપણે બધા ફાર્માસિસ્ટોને થોડો સુધારવાનો મોકો આપીએ છીએ, તો ભવિષ્યમાં પણ આ બધા ફાર્માસિસ્ટ પોતાના રેન્ટ આઉટ લાયસન્સ પાછા લઈ લેજો. નહીતર એમાં પણ આપણે નવી સિસ્ટમ હમણાં ટૂંક સમયમાં લોન્ચ કરવાના જ છે, જેમાં તમને સ્માર્ટ કાર્ડ આપવામાં આવશે. ચીપ અને બારકોડ બંને એમાં ઇન્સ્ટોલ હશે. અને આ એનાથી ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલ અને ફૂડ એન્ડ ડ્રગને ખબર પડશે કે તમે ક્યાં ફાર્માસિસ્ટ તરીકેની ડ્યુટી નિભાવી રહ્યા છો અથવા તો કઈ તમે પ્રોફેશનલ સર્વિસમાં એંગેજ છો. તો પછી આજ નહીં તો 6 મહિનામાં તમારું જે પણ વસ્તુ છે એ ઓપન થઈ જવાનું છે. તો અત્યારથી તમે સુધરી જાઓ તો જેથી કરીને પાછળથી કોઈ ગુનામાં સંડોવાનું ના આવે. કારણ કે જ્યારે બી તમે આઉટ ઓફ ઇન્ડિયા જશો ત્યારે આ લીગલ ઇસ્યુ હશે. ઇવન કોઈને કેનેડા જવાનું હોય અને લીગલ આવા ઇસ્યુ થશે તો ગુજરાત ફાર્મસી કાઉન્સિલ ગુડ સ્ટેન્ડિંગ સર્ટિફિકેટ આપતી હોય છે. એટલે તમારું કેરેક્ટર સર્ટિફિકેટ જે કેનેડા, ઓસ્ટ્રેલિયા, દરેક ફોરેન કન્ટ્રીમાં જરૂર પડતું હોય છે. તો એમાં તમને કાઉન્સિલ કોઈ દિવસ પછી તમને કેરેક્ટર સર્ટિફિકેટ આપશે નહીં અને તમે ત્યાંના ફાર્માસિસ્ટ થઈ નહીં શકો. એટલે આપણા સર્ટિફિકેટની આટલી એક વેલ્યુ છે એ આપણે બધાએ સમજવી જોઈએ. બીજું છે કે હમણાં કેન્દ્ર સરકાર દ્વારા ગુજરાતમાં એક સિલેક્ટ કરવામાં આવ્યું છે કે એપીઆઈ પાર્ક અને મેડિકલ ડિવાઇસ પાર્ક. એપીઆઈ પાર્ક ભરૂચ અંકલેશ્વર પાસે અને રાજકોટ પાસે મેડિકલ ડિવાઇસ પાર્ક 1000 કરોડ બંનેને ફાળવેલા છે ડેવલપમેન્ટ માટે. તો આવનારા સમયમાં જે પણ ગુજરાત, ઇવન તમે ફોરેનમાં જોશો તો અત્યારે ફાર્મસી ઓફ ધ વર્લ્ડ ઇન્ડિયા બની ચૂકી છે, એમાં પણ ગુજરાત નંબર 1 છે. તો એમાં બી આપણે એ સ્ટેટના ફાર્માસિસ્ટ હોવાનો ગર્વ હોવો જોઈએ. અને એના માટે તમારે પોતાની સ્કિલ ડેવલપ કરવી પડશે, કાં તો પોતે બિઝનેસ કરવો પડે. 2 ઓપ્શન છે આપણે, કાં તો સારી જોબ કરો અથવા તો સારો બિઝનેસ કરો. તો એમાં બંનેમાં સ્કિલની જરૂર છે. તો આવા જે રિફ્રેશર કોર્સનું આયોજન થાય છે એમાં તમને બંને જ વસ્તુ નોલેજ બી મળશે, તમારી સ્કિલ પણ અપડેટ થશે. અને ઇવન કેન્દ્ર સરકાર દર મહિને નવા ફાર્મા પોલિસી બદલતી રહે છે. નેશનલ ફાર્માસ્યુટિકલ પ્રાઇસ એનપીપીએ છે, ફાર્મા મિનિસ્ટ્રી છે, દર મહિને નવા કંઈક દવામાં અથવા તો પોલિસીમાં સુધારા આવતા હોય છે. ઇવન ડ્રગ કંટ્રોલર ઇન્ડિયા પણ નવા સુધારા લાવતા હોય છે. તો જ્યારે દેશના બધા રૂલ્સ રેગ્યુલેશન બદલાતા હોય ત્યારે ફાર્માસિસ્ટે એન્ડ ટુ એન્ડ સુધી જે પણ રૂલ્સ નવા આવ્યા અથવા તો શું નવું આવ્યું છે એની માહિતી હોવી જોઈએ. નહીતર તમે 50 નહીં 100 વર્ષ પાછળ જતા રહેશો પછી તમારું આઉટડેટેડ, અત્યારે તો તમે સર્ટિફિકેટ રેન્ટ આઉટ આપો છો પછી એની પણ વેલ્યુ ભવિષ્યમાં નહીં રહે. તો એ પણ એક સિરિયસ મેટર છે. અને જે એપીઆઈ પાર્ક અને મેડિકલ ડિવાઇસ પાર્ક જે ડેવલપ થવાનું છે એમાં આપણા ફાર્માસિસ્ટ મિત્રોએ કેમનો બિઝનેસ કરવો અથવા તો કેમનું જોઈન થવું એના માટે પણ ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલે એક લાસ્ટ 15 દિવસ પહેલા વેબિનારનું આયોજન કરેલું હતું. એમાં પણ સારા ફાર્માસિસ્ટોના જે મગજમાં થોડા ઘણા પ્રશ્નો હતા એ આપણે સોર્ટ આઉટ કર્યા હતા. અને હજુ એમાં પણ કોઈને પ્રશ્નો હોય તો ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલમાં પર્સનલી આવીને અથવા તો મેલ દ્વારા જાણ કરીને આપણે ફ્રી ઓફ કન્સલ્ટિંગ પ્રોવાઈડ કરીએ છીએ. કોઈને નવો બિઝનેસ ચાલુ કરવો, કોઈને નવો મેડિકલ સ્ટોર કરવો હોય, ઇવન કંઈ પણ ફાર્મા રિલેટેડ નવું ચાલુ કરવું હોય તો ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલ ફાર્માસિસ્ટોને સપોર્ટ કરવા માટે રેડી છે. કારણ કે અત્યારે આપણા ફિલ્ડમાં નોન ફાર્માસિસ્ટો વધી ગયા છે, ફાર્માસિસ્ટો ઓછા છે. તો મેક્સિમમ ફાર્માસિસ્ટો આ બિઝનેસમાં આવે એવું ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલનું એક પ્રયત્ન છે, એમાં તમે બધા મદદ કરશો. બીજું છે કે આપણે જે ડ્રગ ઇન્ફોર્મેશન સેન્ટર અત્યારે ડેવલપ આપણે ઘણા વર્ષોથી ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલમાં ચાલી રહ્યું છે, અને એની માહિતી દરેક કોલેજ, દરેક ઇવન નાના સામાન્ય પબ્લિક પણ ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલમાં દવાની માહિતી વિશે પૂછતા હોય છે, બાય મેલ દ્વારા, બાય રૂબરૂમાં, બાય ટેલિફોનિક. આપણી વેબસાઇટ ઉપર એની ઇન્ફોર્મેશન પણ આપણે આપેલી છે ડ્રગ ઇન્ફોર્મેશન સેન્ટરની. તો તમને ક્યાંય જોબ કરતા હોય, મેડિકલ સ્ટોરમાં હોય, ઇન્ડસ્ટ્રીમાં હોય, તો હોસ્પિટલમાં હોય, પરંતુ જો તમારી પાસે નોલેજ નહીં હોય દવાનું, તો પછી તમને ખબર નહીં પડે. તો ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલે કોઈ પણ ફાર્મસિસ્ટનો પ્રશ્ન હોય દવા રિલેટેડ, કારણ કે દવા એટલી બધી મોલેક્યુલ્સ આવી રહ્યા છે, તો દરેક ફાર્મસિસ્ટને બધાનું નોલેજ હોય એવું કમ્પલસરી નથી, પરંતુ ના હોય એ ભૂલ નથી, પરંતુ આપણે ના મેળવીએ એ બહુ મોટી ભૂલ છે. તો એના માટે ફાર્મસી કાઉન્સિલે ડ્રગ ઇન્ફોર્મેશન સેન્ટરની સ્થાપના કરી છે. એમાં પણ તમને વિધિના તમારો જે પણ સમય, તમને ઇમરજન્સી હોય તો વિધિના 2 અવર્સ, અગર ના હોય તો વિધિના 24 અવર્સ તમને સાયન્ટિફિકલી તમને માહિતી મળશે. કારણ કે ગૂગલ ઉપર તો બધી માહિતી મળે જ છે ફ્રી ઓફ કોસ્ટ, પરંતુ જે સાયન્ટિફિકલી જે પ્રૂવન છે એ માહિતી તમને ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલ પ્રોવાઈડ કરશે. બીજું છે કે ઘણા ગવર્મેન્ટ ફાર્માસિસ્ટ મિત્રો જ્યારે અમે કાઉન્સિલમાં આપણે આવ્યા ત્યારે કમ્પાઉન્ડર શબ્દ ફાર્માસિસ્ટ માટે યુઝ થતો હતો, ઘણા ડિપાર્ટમેન્ટ કરતા હતા. તો એના માટે બી અમારા કાઉન્સિલના જે મેમ્બર છે હિતેશભાઈ ભુરિયા, એમને 1.5-2 વર્ષ સરકારના એક ડિપાર્ટમેન્ટથી બીજા ડિપાર્ટમેન્ટ ત્રીજા ડિપાર્ટમેન્ટ, કારણ કે ફાર્માસિસ્ટ ઉપર લગભગ 3 ડિપાર્ટમેન્ટ અલગ અલગ જગ્યાએ લાગતા હોય છે. આ 3 ડિપાર્ટમેન્ટ વાળા કોઈ પણ જવાબદારી લેવા તૈયાર ન હતા. પછી આપણે થાકીને ગુજરાત હાઈકોર્ટમાં કેસ કર્યો, અને એ કેસમાં આપણે વિધિના 6 મહિનામાં તો એ લોકો ઇન્ટરનલી સર્ક્યુલર બહાર પાડી દીધો કે ભાઈ કમ્પાઉન્ડરની જગ્યાએ જુનિયર ફાર્માસિસ્ટ યુઝ કરવું જોઈએ. પરંતુ જે આપણા હાઈકોર્ટના એડવોકેટ છે એમને કીધું કે ભાઈ નહીં, આપણે આ કોર્ટ થ્રુ લેવું જોઈએ. તો આપણે એની વેટ કરીને બીજા 3-4 મહિનામાં કોર્ટે લેખિતમાં આપણને ઓર્ડર કાઢી આપ્યો કે ભાઈ સરકારને, કે ભાઈ આ તમે કમ્પાઉન્ડરની જગ્યાએ જુનિયર ફાર્માસિસ્ટ યુઝ કરો. એમ આપણી જે એક્ટિવિટી છે બધા ગવર્મેન્ટ ફાર્માસિસ્ટોના સહયોગથી આપણે એક જે આપણું ગૌરવ કહેવાય કે ભાઈ જે વર્ષોથી નતા મેળવી શકે એ આપણે ફાર્માસિસ્ટ તરીકે મેળવી શકે. હવે જે સમય છે, ઘણા ફાર્માસિસ્ટ મિત્રોના ઘણા એવા પ્રશ્નો છે જ કે ભાઈ અમારો પે સ્કેલ નીચો છે, ભાઈ ફાર્મસીની કેડર નથી. તો આ બધા પ્રશ્નોના પણ આપણે સોર્ટ આઉટ કરીશું. એનું પણ પત્ર વ્યવહાર સરકાર સાથે ચાલુ છે. એમાં પણ જો તમે બધા સપોર્ટ કરશો તો એમાં પણ આપણે આગળ વધીશું અને સારી રીતે મેળવી શકીશું. પણ આ વર્ષો જૂની, અમે તો અત્યારે પણ જે વર્ષો જૂના ફાર્માસિસ્ટ મિત્રોએ કોઈ દિવસ સરકારમાં રજૂઆત જ નથી કરી, કોઈએ કંઈ કામ જ નથી કર્યું. પરંતુ હવે જ્યારે આપણે કામ કરીએ છીએ ત્યારે બીજા 50 લોકો પથરા નાખવા વાળા હોય છે, આપણા જ ફાર્માસિસ્ટ મિત્રો હોય છે. તો આવા લોકોને કોઈ ઇગ્નોર કરો, કારણ કે તમારી જોડેથી 1 રૂપિયો કાઉન્સિલ લેવા માંગતી નથી. અમે કાઉન્સિલ કોઈ કોર્ટ કેસમાં બી તમારો રૂપિયો નથી લેવા માંગતી. તો આવા ખોટા લોકોને માર્કેટમાંથી ઉખાડી ફેંકો અને સારી રીતે આપણે કાઉન્સિલની સાથે રહી, કારણ કે કાઉન્સિલ તમારી છે, તો રજિસ્ટર ફાર્માસિસ્ટ તમે છો, તમારી સંસ્થા છે, તો આપણે સાથે મળીને કામ કરીશું ફાર્માસિસ્ટ માટે, તો 100% આપણે સારી રીતે કામ કરી શકીશું અને ગવર્મેન્ટની કેડર પણ આપણે મજબૂત કરીશું. ને એના માટે બી આપણે બધા મેમ્બર પ્રયત્નરૂપ જ છે. ઇવન આપણે જે બોગસ ફાર્માસિસ્ટ, એકચ્યુઅલી મારું થોડું એટલા બધા આપણે જે કાઉન્સિલમાં આયામો શરૂ કર્યા છે એટલે હું થોડી વધારે ટાઈમ લઈશ, પરંતુ તમને આવું કાઉન્સિલના કામ વિશે જાણકારી આપવી જરૂરી છે. એક તો આપણે ઘણા એવા ડિપ્લોમા ફાર્મસિસ્ટ, તમને ખ્યાલ હશે કે ઘરે બેઠા લોકો ડિપ્લોમાની ડિગ્રી લેવાનું ચાલુ કરી દીધું છે આમાં, કારણ કે ફાર્મસિસ્ટની અછત થઈ ગઈ છે ને. 72,000 ફાર્મસિસ્ટ તો છે જ આપણા ગુજરાતમાં, પરંતુ બધા અમુક ઇન્ડસ્ટ્રીમાં છે, અમુક હોસ્પિટલમાં છે, ઇવન અમુક લોકો ફોરેનમાં છે. તો ઘણા એવા લોકો જે મેડિકલ સ્ટોર વાળા છે અથવા તો જે ડોક્ટરો છે, જેમનો પોતાના હોસ્પિટલ્સ છે, નાના મોટા ક્લિનિક છે, તો એ લોકોએ પોતાના વાઈફ અથવા તો પોતાના નાના બ્રધર અથવા તો મોટા બ્રધરને, જે લોકો ફાર્માસિસ્ટ નતા, એ લોકોને ફાર્માસિસ્ટ બનાવવાનું આખું કૌભાંડ જે ચાલી રહ્યું હતું દેશ લેવલે, એમાં ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલના મેમ્બર ડોક્ટર CN પટેલ સર અને હું અને ડોક્ટર નવીન શેઠ સાહેબે ફાર્મસી કાઉન્સિલ ઇન્ડિયામાં અમે બહુ એની રજૂઆત કરી. અને જે બોગસ કોલેજો કામ કરી રહી હતી રાજસ્થાન અને પંજાબની, એમાં આપણે કેસ CID માં પણ આપ્યો છે. અને લગભગ 80 ની અરાઉન્ડ લગભગ ફાર્માસિસ્ટો જે 50 વર્ષથી મોટા હતા એ લોકો આટલી એજે 12 સાયન્સ ક્લિયર કર્યું અને પછી ડિપ્લોમાની ડિગ્રી લઈ આવ્યા. એટલે તમે જોઈ શકો છો કે આ કેટલા વર્ષોથી આ બધું ચાલતું છે, પરંતુ આપણે આવ્યા પછી આ બધું આપણે સોર્ટ આઉટ કર્યું અને જે લોકો જવાબદાર હતા એ લોકોને આપણે આમાંથી રિમૂવ પણ કરેલા છે. અને અત્યારે સીઆઈડી ની તપાસ ચાલે છે અને એ લોકો 100% કોલેજો તો આપણે બેન કરાવી દીધી છે ફાર્મસી કાઉન્સિલ ઇન્ડિયા થ્રુ કે આ 3 કોલેજો કોઈ નવા એડમિશન નહીં કરી શકે અને નો એડમિશન ઝોનમાં મુકાવી દીધા છે. એટલે આપણા જો ગુજરાત લેવલની જે મહેનત છે આખા ઇન્ડિયા લેવલે એનો પડઘો પડ્યો અને બાકીના જે બોગસ ફાર્મસી પ્રેક્ટિસ કરતા હતા આ રીતે બોગસ ડિગ્રી આપવાનો એ લોકોએ આ વખતે એડમિશન 0 થઈ ગયા છે, એ લોકોના નહીવત થઈ ગયા છે. પરંતુ હજુ બી અમારા ધ્યાનમાં અમુક કોલેજો છે જે લોકો ગુજરાતના સ્ટુડન્ટોને લલચાવીને લઈ જાય છે, એના પર ભાઈ અમે પગલા લેવાના છે. કારણ કે લોકો ઘરે બેઠા ડિગ્રી તો ઘણા લેતા જ હોય છે. આવા જો તમારા ધ્યાને આવ્યા હોય, કાઉન્સિલે રજીસ્ટ્રેશન ગ્રાન્ટ કરી દીધા હોય રજિસ્ટ્રાર દ્વારા, તો આવા કોઈ તમારા ધ્યાને આવે તો એની માહિતી પણ ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલને આપજો, તો એની પણ અમે તપાસ કરીશું. તો આ રીતે બધું આપણે બોગસ ફાર્માસિસ્ટોને પણ નીકાળ્યા છે. અને મોસ્ટલી આ બધા મેડિકલ સ્ટોર વાળા લોકો જે જૂના છે એ લોકો આ બધું કરતા હોય છે, કારણ કે કઈ હોય નહીં એટલે આ બધું અમુક લોકો એજન્ટ પણ બની ગયા છે આમાં. બીજું છે કે ઈ ફાર્મસી, અત્યારે બધા રિટેલરનો એક મોટો પ્રશ્ન છે કે ઈ ફાર્મસી, ઓનલાઈન ફાર્મસી. રિલાયન્સ આવી ગયું, એમેઝોન આવી ગયું, ફ્લિપકાર્ટ આવી ગયું. આજ સવારે મેસેજ આવ્યા કે 1mg વાળાને ફ્લિપકાર્ટ વાળાએ લીધા. એટલે આ બધા ઓનલાઈન પ્લેયરો અત્યારે બહુ દેશવ્યાપી લેવલે વર્લ્ડ વાઇડ આવી રહ્યા છે. અને ડિજિટલમાં જે યુગ અત્યારે જે છે એમાં આપણે કંઈ બધાને રોકી તો નહીં શકીએ, પરંતુ આ બધામાં ફાર્માસિસ્ટનો ઇમ્પોર્ટન્ટ રોલ કેમનો રહે? કારણ કે મેં તમને પહેલા જ કીધું, આપણે કોમર્શિયલી નથી થવાનું, પ્રોફેશનલી થવાનું છે. તો આપણે ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલમાં લગભગ 1.5-2 વર્ષ પહેલા આપણે બધા ગુજરાતના અલગ અલગ જિલ્લાઓમાંથી, એસોસિએશનમાંથી અને ગવર્મેન્ટમાંથી અને દરેક જગ્યાએથી બધા રિપ્રેઝેન્ટેટિવને બોલાવેલા અને એક આનું મિટિંગનું આયોજન કર્યું. આ મિટિંગમાં જે પણ પોઝિટિવ મુદ્દા હતા કે જેમાં ફાર્માસિસ્ટનો ઇમ્પોર્ટન્સ રહે એ રીતે રૂલ્સ બનવા જોઈએ. અને આપણે આ રજૂઆત કેન્દ્ર સરકારમાં કરી, ઇવન ફાર્મસી કાઉન્સિલ ઇન્ડિયાએ આમાં આપણને સપોર્ટ નહોતો કર્યો એ સમય, તો પણ આપણે ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલે કેન્દ્ર સરકારની મિટિંગમાં હાજર રહીને એની રજૂઆત કરી ટેકનિકલ મુદ્દા સાથે, ફાર્મસી એક્ટ અને PPR 15 સાથે. તો એમાં સરકારે આપણી વાત ધ્યાને લીધી અને હજુ સુધી કોઈ રૂલ્સ બનવા દીધા નથી. પરંતુ રૂલ્સ તો બનવાના છે, બધા મોટા પ્લેયર છે, બનશે ખરા, પણ હજુ પણ સરકારે આપણી પાસે પોઝિટિવ જેમાં ફાર્માસિસ્ટ અને રિટેલ મેડિકલ સ્ટોરનું એક્ઝિસ્ટિંગ રહે એ રીતે રૂલ્સ બને એ દિશામાં કેન્દ્ર સરકારને આપણે વિઝન આપ્યું છે. અને લગભગ બધા જેટલા બી ઓનલાઈન પોર્ટલ વાળા છે ખાલી પોર્ટલ જ રહેશે, કોઈ દવાનું ડિસ્પેચ કરી નહીં શકે. દવાનું ડિસ્પેચ જસ્ટ મેડિકલ સ્ટોર વાળા જ કરી શકશે, તો ફાર્મસી સ્ટોર જ કરી શકશે. એવા રૂલ્સનું આપણે આયોજન ફ્રેમ થાય એ રીતે પ્રયત્નો ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલના મેમ્બરો દ્વારા કરવામાં આવ્યું છે. એટલે આ બધી રજૂઆતો આપણે છે નાની રજૂઆત, પણ બહુ મોટો ફાર્માસિસ્ટનું ઇમ્પોર્ટન્સ રહે એ રીતે આપણે બધા પ્રયત્નો કર્યા છે. અને ઘણા એવા તમને ધ્યાને છે કે બી મારે આમ તો ના કહેવું જોઈએ કે બી ઘણા એવા જે લેડીઝ ફાર્માસિસ્ટ હોય છે ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલમાં, લગભગ બહુ બધા લેડીઝ ફાર્માસિસ્ટ રજીસ્ટર થયેલા છે. એ કાં તો હાઉસવાઈફ હોય છે અથવા તો પાર્ટ ટાઈમ માટે પોતાના લાયસન્સ રેન્ટ આઉટ કરતા હોય છે. એક મોટી ચેન જે આપણા ગુજરાતમાં એક્ઝિસ્ટિંગ છે, એપોલો ફાર્મસી. હવે એપોલો ફાર્મસી જો આપણા ફાર્માસિસ્ટોના લાયસન્સ રેન્ટ ઉપર ચલાવતી હોય તો, અને એ પણ આપણા તમે જોતા હશો, તમે માર્ક કરજો, એપોલો ફાર્મસીમાં કેટલી છોકરીઓ ફાર્માસિસ્ટ તરીકે ડ્યુટી કરતી હોય છે. હા, 10% કરતા હશે, પણ 90% ડ્યુટી નથી કરતા હોતા. આ બધા લોકોને પણ આપણે પકડ્યા છે અને લગભગ 1-2 સ્ટોર તો આપણે બંધ પણ કરાવ્યા છે એપોલોના. તો આપણે આ બધા ફાર્માસિસ્ટ મિત્રોમાં જે લેડી ફાર્માસિસ્ટ છે એમને પણ રિક્વેસ્ટ છે કે તમે જોબ કરો એનું ના નથી, પરંતુ આ રીતે રેન્ટ આઉટ કરશો તો બાકીના જે લોકો છે, જે એમની એક ફેમિલી ઉપર 4 લોકોનું ગુજરાન ચાલતું હોય છે, અગર આપણે રેન્ટ ઉપર આપી દઈએ તો આપણે 4 લોકોનું ઘર બગાડી રહ્યા છે. તો આ વસ્તુ ના કરવી જોઈએ. એની પણ એક મેસેજ ગુજરાત રાજ્ય ફાર્મસી કાઉન્સિલને હું આપને કન્વે કરવા માંગીશ. તો આ બધા જ કાઉન્સિલના પ્રયત્નો છે અને હજુ પણ તમારા કોઈ સજેશન હોય તો ફાર્મસી કાઉન્સિલને તમે આપી શકો છો. અને પર્સનલી કાઉન્સિલમાં જો તમારે કંઈ ડિટેલમાં જાણવું હોય તો કાઉન્સિલ પણ રૂબરૂ આવી શકો છો. બાકી 2 દિવસનું જે રિફ્રેશર કોર્સનું આયોજન છે એમાં સારા સ્પીકર પણ છે, અને મેડમ ડોક્ટર હર્ષા મેડમ છે અને બીજા ઘણા એમના સ્ટાફ ફેકલ્ટી મિત્રો પણ સારો. આ ઓનલાઈન કરવું એ ક્લિક કરવાથી ઓનલાઈન નથી થઈ જતું, પરંતુ 15-20 દિવસ એમ મહેનત લાગતી હોય છે. પછી આખું આપણે અત્યારે ઇઝીલી વાત કરી શકીએ છીએ. તો એમના માટે પણ હું આભાર વ્યક્ત કરું છું. થેન્ક યુ. જય હિન્દ, જય ભારત.
Thank you so much, sir, Montu sir, for the information and throughout support for the refresher course. We are heartily thankful to you, sir.
Thank you.
I appeal Engineer Bhikhubai Patel, sir, Chairman of Charutar Vidya Mandal University, for his presidential remarks. Sir, please.
Yes, speakers and registered pharmacists. To know that Indubaka Incovala College of Pharmacy is organizing GSPC Council Food Refresher Course for registered pharmacists. I extend a warm welcome to all the participants and I'm very happy to note that today you all are here. I am sure esteemed speaker will deliver above and beyond your expectation during these 2 days. I would like to say to all the participants that please make the most of what we expert have to say. Update the pharmacist plays a very important vital role in the community healthcare. Interaction with GSPC President will provide you revenue of costs and action which will be benefited to all participants. I wish the speaker and participants have very lively and informative instructions. This is today's refresher course. Thank you. Good morning to your speakers and registered pharmacists. I am pleased to know that Indubaka Incovala College of Pharmacy is organizing GSPC Council Food Refresher.
Thank you, sir, for encouraging us with your words and blessings. Now we are moving towards the plenary sessions. During the sessions, if anybody has any questions, they can drop their questions in the chat box. Attendance link for every session will be given to you during any time of the lectures. So the path of the person may be dark, but it gets enlightened by the light called teacher. Now it's time for our first plenary session by Professor Dr. Joyvardhan K. Patel, sir, Professor and Principal, Department of Pharmaceutics, Nutan Pharmacy College. He will be delivering a lecture on patient counseling. I would like to introduce you, sir. He is obtained from his BPharm degree from BK Modi Government College of Pharmacy, master degree in pharmaceutical technology from the prestigious pharmacy department, MS University Baroda, in pharmaceutical sciences, LLB from Hemchandracharya North Gujarat University, Patan, and doctorate degree from Department of Pharmaceutical Science, USF College of Pharmacy, University of South Florida. At 22, he started a career as a production manager in the Samrat Pharma Laboratories, India, way back in 1996. Currently, he is the professor of pharmaceutics and principal, Nutan Pharmacy College, and dean faculty of pharmacy, Sankalchand Patel University, Visnagar. He has more than 24 years of academic and research experience, has published 230 research and review papers and in international and national journals, and has presented more than 150 research papers at various international and national conferences as author and co-author. Dr. Patel has presented 32 research papers at various international conferences. He is the author of 14 books and 44 book chapters. He has guided 38 PhDs, 104 MPharm students for dissertation work, and 8 students are currently pursuing their PhD under his guidance. He has completed 12 industry and government sponsored research projects. He is the recipient of Fast Track Young Scientist Award by SCRB Government of India, New Delhi, and 19 for 4 lakh rupees. Dr. Patel has received about 186 lakh grants till date from the different government bodies. The total he has received INR 25 millions for the research project or international travel grant or organizing conference grant. He holds one patent and one patent is registered. He visited different countries for the multiple times for research paper presentation and as a resource person. Uh, as a capacity coordinator, he organizes 20 national seminars and conferences to FDPs, to popular lectures on biology and 5 pharmacist refresher course sponsored by different organizing bodies of India. He approved as a PhD guide for pharmacy at Sankal Chand Patel University. He is serving the peer reviewers of 50 well-reputed journals. Dr. Patel is serving as an associate editor, member editorial board of 14 journals. Dr. Patel is recipient of Outstanding Young Indians 2010 award by JCI India, Young Pharmacist Award 2013 by Indian Pharmacy Graduates Association, Young Scientist Award 2014 by Association of Pharmacy Professionals, Principal of the Year 2014 by Award by Research Scholar Hub, very prestigious APTI Young Pharmacy Teacher Award 2014 by Association of Pharmaceutical Teachers of India, Eminent Teacher of Year 2016 by Research Scholar Hub, Fellowship Award by Research Scholar Hub, Distinguished Professor in Pharmaceutics during DST, NST, MIS sponsored Stakeholder Workshop 2019 and Research and Presidential Award and 1 lakh for distinguished contribution in teaching and research by Sankalchand Patel University. Dr. Patel is recipient very prestigious AICTE Visvesvaraya Best Teacher Award 2020 by AICTE DST, Ministry of Science and Technology, Government of India, has published international competitive research base, which is throughout 2009 to 2014. The Dr. Patel has put in honors list of 4th rank of 10 authors in subject area of pharmacology, toxicology, and different branches. So it's our great privilege to have you here today, sir. Thank you for accepting our invitation. Over to you, sir.
Thank you. Please share my slides.
Yes, sir. 1 second.
So good afternoon, all dear pharmacists. Yesterday was the day, the World Pharmacist Day, and we have celebrated in the world.
It is visible, sir?
Yes, yes, please.
Yes, sir.
So by mistake, I have been put the dates wrong. It is 26th and 27th September. For apologized for the same date should be changed, 26 and 27. So by the Gujarat State Pharmacy Council sponsored 2 days pharmacist refresher course, I have been given one topic is a patient counseling. Next. Okay, dear pharmacists, that we know the patient counseling is one of the major duty of the pharmacist rather than dispensing of the drug. As Montubai said, the duty of the pharmacist, we are only dispensing of the drug at the medical store. The process of the purchasing of the drug in our country is like doctor is writing a prescriptions, our caretaker or our relatives or patient himself or our family member to the medical shop, and they are just dispense the drug to the patient. Nothing, no conversion between the patient and the pharmacist. It is more important, the conversion between the patient and pharmacist should be there. So the patient counseling, we can define the providing medication information orally or in written form to the patient or their representative on the direction of uses, advice on the side effects, precautions, storage, diet, or the lifestyle modifications. As we know, pharmacist, at pharmacy store in India, nobody is explaining about how to use the medications. Nobody. Advice on the side effects. There are many side effects or drug food interactions. We have many examples. Examples, that is one is the antibiotic with dairy products like milk or the yogurt that contain the calcium, and drug like ciprofloxacin or tetracyclines. That is that will be a delay or maybe prevent the antibiotic that effectiveness. Then precaution, how to store the drug, diet. Nobody is taking care about, or doctor is only taking care about the what the diet should be taken by the patients. But pharmacists also know which type of the diet should be a need for the patient with the medications. Next. So objective of pharmacy patient counseling is the patient should be recognize the importance of the medication for his or her well-being. As we know, the patient is never be known with the medications, but if we counseling with the patient, then patient is know about the importance of the medications. The working relationship between the patient and the pharmacist will be established. Patient understand the strategies deal with the medication side effect and drug interactions should be improved. There are many examples. Even one I can quote one example of oral diabetic with the alcohol. That is very dangerous, both together, even insulin with the alcohol. Should ensure better patient compliance after the counseling. Patient become an informed, efficient, and active participation in disease treatment and self-care management. So patient himself or herself may be take care of their disease treatment. And the pharmacist should be apparent as a professional and who offer the pharmaceutical care. In this, if the patient counseling may be in India, then the pharmacist value will be there. Next. So patient counseling have three stages. First is the introduction. Second is the process and the issue regarding the manners or the approaches. And the third one is the conclusion. Please, next. In introduction, once patient comes to the or you are start to counseling any patient, before this I have to go with yesterday program. That was the pharmacist day, and the pharmacist stream was transforming the global health. Now transforming the global health, we are talking about the global health, but the pharmacist role should start from their home only first, because pharmacists know where to store their medications, how to store the medication, how to take the medication, when to take the medication, how to avoid two medication together, how to avoid the food with the which food with the medication. Many things we can do at home first, because lifestyle nowadays very typical, and every family maybe or each family have a one patient at home, and they are taking the medication every day. So we have to take care from we have to start this counseling from home first, then family first, then society, then will be comes to the village, then comes to the taluka place, then comes to the district level, then comes to the state level, then comes to the country level, and then comes to the global level. So the challenge for the pharmacist is too much important. So every pharmacist should know about how to do the counseling and what is an important of the counseling. So before the counseling, review the patient record, introduce yourself. I am Patel, I am pharmacist, and I will do your counseling and what is an important of the counseling, please. Then explain purpose of the counseling. Continue, same last slide. Okay. Explain the purpose of counseling to the patient, because no patient is ready for the counseling. If you try to explain, then they will ready to be a listen. Obtain drug related information from the patient, like allergic or use of the herbals. If then you can review it. Review the patient understanding the reason of the therapy. We have to explain to the patient. An example is suppose the diabetic or the hypertension, the therapy need for the whole life. So we have to explain that these tablet or these dosage form you have to take for the how much longer time. Okay, and review any actual and or possible concern of the problem with the importance of the patient. We can do next. Then issue regarding the manner or approaches. if we are starting the counseling or counseling we start in India, we know that the mechanism of the dispensing of the drug, like I earlier said, the doctor is writing the prescriptions. We at the medical shop we are buying the medications, and the procedure is nothing. If we any medical shop, we can see without the we never ask the pharmacist is present or not. Even being a pharmacist, we study the pharmacy. There are want to say that is about 70,000 registered pharmacies in Gujarat. So even we are not bother about where we are buying the medication, the pharmacist is there or not. There are 2 or 3 taberias are there in medical shop everywhere I've been seen, and they are providing medication to you, and medication are provided in the blue or yellow or black polythene bag by staple. We have to take in morning or evening, or that will be taken after meal, or nothing should be written on the prescription, and nobody is taking care about the how to take the medications. Okay, so when we counsel with the patient, we must need be use the language that patient understand. If we are at the village, that definitely people or the patient never be understand the English or the maybe Hindi, but we have to be explain him in the our own languages. Use the appropriate counseling tool. Means if you have the paper and pencil, if you explain on the paper and pencil, that will be better. You can utilize such type of the counseling tool, or present fact and concept in simple word in the logical order, because patient can't understand what are the drugs. Drugs is we know what is the definition of the drug. Nowadays, you in every or each newspaper, the headline is the actress or the actors with the drugs. So what is the difference between the what we are dispensing or we are distributing the drugs, and that the drugs that are used by the our film industry people? Both are similar. We can, but one drugs or the drug what we are distributing being a pharmacist are for the patient. This is drugs that cure the patient health, while other drugs that is a misuse in the misuse by the other people. So pharmacist's role, it is a dispensing or not of the drug, but we have to be very taken care about which type of drug we can distribute to the patients. Okay, so we have to and we have to explain in simple words, because patient is very unknown with the drugs. Use open-ended question. Patient may be asked any question, you must have to be explained in logical order. Next. So in conclusion, after the our counseling, we must have to verify the patient understanding by mean of the feedback. We have to say what you understand after my counseling. If you are satisfied, then you have to continue, or then the counseling part is over. Summarize by highlighting the key point. If patients have the prescription with the 10 or more than dosage form described by the or prescribed by the doctors. So if you explain about the 1, 2, 3, and then 10, maybe he forgot about the previous one. So we have to summarize in a point wise to the patient, and give an opportunity to the patient put forward and deal with. So that will be a any chance, any suppose patient forgot about the previous one, you have to be a take help. You have to give the help to the patient for planned follow-up. Means patient comes to you anytime, anywhere. Next. So who need and when need the counseling? It is really that the pharmacist counseling patient on the new or the refill prescription. New prescription is the first prescription provided by the doctor to the patient, and that will be comes through the pharmacist. And refill prescription considers any change of the medication by the doctors, or addition of the medication or removal of the medication. In this case, that definitely we need the counseling. Next. The patient have the more specified number of the medication need the counseling more. The patient have visual hearing problems, and the pediatric patient is also need the more time counseling, because the children or the child are the very naughty people, and we have to give some thing that you picture. You can see the doctor or the pharmacist or the doctor is providing the one apple to the children to explaining about his or her disease. Next. So the function of the patient counseling, if we can say, it is include the better patient understanding for that illness and role of the medication in treatment. Earlier I told, if the medication taken by the patient, he or she don't know what happened inside into the body, and patient need to be suppose we have the pain or the headache, and we can understand the drug like the paracetamol or the diclofenac we taken, and we observed within 15 to 20 minutes should be a headache should be down, or pain should be less. But it is not possible. It is depend on their illness, their therapeutic effectiveness. So we have to be understand, we have to say to the patient, it is need 1, 2 or 3 days for the drug effect. Then improve medication loyalty is also an important part in the India. So how we can consider the drugs or the medication what we are distributing or the dispensing to the patient are loyal, that nobody can change or nobody is challenged. The all drug are the loyal, because in India the drug market or the drug what distributed, they are not 100% loyal. So we must have to be taken care. The company, those who are preparing, those who are working with the company, those who are working with the quality assurance department, all drugs should be in their quality criteria. More effective drug treatment, reduce incidence of the adverse effect, unnecessary health cost. Unnecessary healthcare cost, that is in India, it is too much high, because doctors are only sometimes trying on the patients. They are writing few another drugs. It is if it is unwanted for the patients. So pharmacists can know these drugs are not an important and will be not helpful to the particular patient, so that should be removed from the prescriptions. But this authority is not given to the Indian pharmacist. This authority has been given to the pharmacists, those who are working at the abroad country like the USA, Australia, Canada, or the United Kingdom, like developed country. And why the pharmacist value at the USA is higher than the USA in higher than the India? Because of they are studied here. Your friend, my family member, or your family member maybe studied your classmate. Even they went to USA, and then they pass the examination and they become a pharmacist. But now they have about $50 per 1 hour. They are getting pay, and here the pharmacists are the retail pharmacists are maximum 15 to 20,000 maximum I am talking about. So why it is different? One pharmacist, those studied in India, your classmate are getting one day 15,000, and you are getting 15,000 in 30 days. The difference is nothing, but different is the clinical related data. They know their knowledge is different than us, because there are the million of the molecules available into the market, and the market molecule, each molecule have that different clinical data, such as the therapeutic effects, such as the adverse effect, such as the interaction, such as the many things. But each molecule detail we must have to be in our knowledge when we dispense the drug to the patients. Very important. There are a few questions. I will give your answer to the audience. Don't worry about it. Then improve the quality of the life of the patient and improve the professional relationship between the patient and pharmacy. Another point I have to notice here, in abroad, there is pharmacy working at the CVS or other, even their private medical shop. If you have the number of many times of the prescription filling, that pharmacists earn more and more. Means each prescription for each prescription filling, the pharmacists earn $10 to $15 or the pound for their credit. So if you have the good relation with the patient, means the patient will comes to you because it is dependent on the professional relationship. If you explain well, then surely the patient will comes to you next time in your pharmacy store. Otherwise, it is very difficult to be come there if you dispense the not proper the drug to the patients. Next, the patient who need always counseling, the listed are the confused patient and their caregivers. Confused patient that definitely need the counseling for more and more time. The patient who are sight or hearing impaired, those who are not well educated. Patient who profile shows change in medication or dosing. If the medication not work on the patients and doctor are prescribed the change of the medication, that time we pharmacists have the role is better than the doctors. We have to explain because it needs some time to be set the medication into the body. Then new patient or those who are relieving the first time or transfer, the children and parents receiving the medication, then patient receiving the medication with special storage requirement and complicated directions such as a suppository, patch and inhaler. I have this example I have kept here because the patches or the patch, the basic most and important example is the diclofenac. We people are used diclofenac tablet or paracetamol tablet like chana and mamra throughout in our life. We are taking small pain, we are taking the diclofenac or paracetamol. We are not to get ready, we are not giving time to body to react with the pain, and body itself should be remove the pain by their immunity. We are never be giving a chance. So patch, the diclofenac example is the orally diclofenac that we know it is very dangerous drug. If it is taken an empty stomach, then it is a very dangerous that shows the hyperacidities. But patch of the diclofenac, if stick on the skin of any layer of the skin, and the drug is released and no adverse effects shows with the patch. So such type of the how to stick the pitch, where to stick the pitch, how to use the suppository, how to use the inhaler. Patient don't know about the inhaler. So we have to explain how to use the inhaler properly, how to take the breath after the inhale the drugs into the inhaler. So that is an important part of the pharmacy to explain the use of the direction of the use of the medications. Also, we have to be explain about the special storage requirement. I've been seen many times in uh, even in Gujarat or in my taluka place or the city place, the syrup is stored at the refrigerator many times. Now, pharmacists, we know because we are not stored, because we understand about the stability. But many people, our even we can see in our neighbor, they are stored the syrup at the refrigerator. We know what happened with syrup when we store in the refrigerator. So where to store the medications? That is an important also task. And in even pharmacist houses also the medication stored on the top of the refrigerator. This is also dangerous for the medication because top of temperature of the top of the refrigerator is about more than 40 degrees. So that is a decomposition of the medication chances are more and more. Next. So patient who need the counseling at certain intervals, means no need for the whole time, like patient like the asthmatic patient, diabetic patient, then patient who are mentally ill, the patient with the skin complaint, misusing drug, etc. These patients need the counseling at certain interval by 1 month, by 2 months, by 3 months, not need for every time, even changing the prescription. Next. The counseling area is more important. As we know, as India, there is a no counseling area. Yes, it is defined by any body, but in a foreign country that is there, separate glass cabin for the patient and the pharmacist can sit. There are 2 seats, and they can counseling at and private, not in public, because patient may be shy to say something in not in public, but they can explain or they can uh, maybe say to the pharmacist everything in their personal. So it should be a confidence, security should be maintained there. So the we need to be a counseling area separate at the medical shop. Next. The communication skill for effective counseling, that we know that are two type of the counseling process. User are verbal and non-verbal communication skill. The verbal communication skill, we can include the language that earlier I told you. The language we have to be utilized what the patient can understand. Gujarati, so we have to explain in Gujarati. Hindi, so Hindi we have to explain to Hindi, or English, so we have to explain it to Hindi. Okay, then tone should be a very proper. It should not be loud, not should be a, that means tone is like there are many examples of the tone, so it should be a very proper. Volume also should be a very proper and very effective, that patient can listen you well, not too much loudly you have to speak. Speed should be a very normal, not to be a slow, not should be a very fast. It should be a very proper. Then non-verbal communication should be a close net. You don't should be a you don't should be a sit very close to the patient. It should be a distance between the patients and not should be very far, so patient can't understand your expression also. The facial expression is also an important. When we meet to any patient, we must have the work as a professional. Professional means we are performing our duty 8 hours only. Rest of 16 hours we have to give to our family. So these 8 hours are very crucial for every or each pharmacist. We have to be a professional. Yes, definitely. There are many problems at family or maybe in society, maybe in your workplace. But once you are doing the counseling, your mind should be very clear. Your face should be very that smiley, not like khichdi and curry. If you have any problem at the home or the fight with the wife, and then we are trying to be counseling, it is very difficult. Okay, so what happened at family, put at the home, and comes when we are professional, we have to be understand what is a professionalism. Next. So quality of good counselor may be first be a good listener. When you have the knowledge, you can provide the knowledge. So we have to be a good listener first, be a flexible and unbiased or the neutral. Don't be a bias with anyone. I have the one good example, that is, suppose young pharmacist. If girl comes for the counseling, maybe pharmacist providing the more time for the beautiful girl. If the aged people and comes to for the counseling, okay, kaka, it is okay, you're finished. Miss, we are just hurry to finish with the uncle or the auntie. Not like, don't bias between the beautiful girl and the old patient. Should be a similar. So counseling given should be a very proper and unbiased. Be open-minded. Patient may be asked any question, you should be ready to answer. And communicate confidently. If you know, then you can only explain. If you unknown with any complicated requirement asked by the patient, if you don't know, then don't say today. Maybe you can say after, later on. Okay, so communication, the confidence is also an important. Next. So step during the counseling, the two steps are there: preparing the session and opening the session. And successful counseling require the preparation of the counseling, the select the suitable place, schedule the time, then notify your subordinate. They will be prepare your advance for your and patient, organize information for the patients, then outline the counseling session component, what we are going to be counsel to the patient, plan your counseling strategy, and establish right atmosphere. Then patient will there, and then we can start the counseling. So this will be a step for the counseling. Next. So counseling content, it is a much more important is that during the step, the pharmacist explain the patient about his or her medication and treatment regime. Also, we have to be explain about lifestyle change such as diet or the exercise may be also be discussed. Have you seen any time we buy the or the brought the medication and pharmacists have to explain, don't take such type of food with your medication? Nobody. Only doctor is prescribe the drug and giving the advice to the patient, always take khichdi and doodh, rice and dal, etc. Nobody is talking about the proteins or other nutrients needed for the patient, for the patients. That is much more important. So nutrients requirement, if need, we have to explain to the pharmacist. Next. The topic mostly commonly covered should be a name and strain of the medications. We have to explain to the pharmacy reason why the prescribe the drug and how it will be work. Earlier told you, suppose sublingual tablets is prescribed by the doctor. Patient never be understood if I put on the below the tongue and how it will be help. It will be work, but we have to explain. If you put on the tongue, will be goes directly to the blood circulation and effectiveness is higher than the oral. We have to explain how to take the medication is also important. There are many people is never be utilize how to take the medication. Even I have been seen the dispersed tablet are they are taking orally, or dispersed like Disprin or the aspirin type of tablet they are taken orally. Okay, it is no problem, but if we have to give the proper effectiveness, should be a disperse or despair that should be in solution form, then take that will be a best or the higher effect. We have to explain. Then expected duration of the treatment, we have to understand. The example are the antibiotics. Suppose many times the patient are buying the antibiotics that the doctor is prescribed for the 3 days. If they are buying for 1 or 2 days antibiotic, or they're taking only 4 dose instead of the 6 doses. That is commonly we have to explain such type of the only 4 doses as antibiotic that is unused, that will be not effective. So we have to explain that need compulsory 3 days dose for the treatment for the antibiotic. Then expected benefit of treatments, we have to explain. Possible adverse effect, yes, definitely affect, because drugs are need and maybe adverse effects comes, we have to explain. You will have some nausea, some vomiting after these medications, so take care of about the adverse effect if it is there. Possible medical or directly interaction, advice on correct dose, correct storage, the minimum time duration required to show the therapeutic effect, that explain about any medications. If you take on paracetamol and if fever or the higher temperature, that definitely need about 6 hours to reduce the temperatures. We have to explain them. Then what to do if dose is missed. This is also an important part to counsel to the patient, or we have to explain to the patient if you miss the dose, how to take the medication later on. Okay. Then special monitoring requirement, blood test for the diabetic patient or etc. Next. So the overall conclusion of the presentation is, at least we can say the patient counseling is a part of the parcel of the good medications. The good counseling can provide the patient to take her or his medication. It is not we are providing medication, but it is we are providing the parcel of the good medication. It is the most important rather than dispense of the drugs. Dispense of the drug, that can be a do by the taberia what available at the medical shop. They can do better than us. They know there are even 1,000 molecules are there. They can find out the molecules within 2-3 minutes. And then they know about the many names of the medication. Even pharmacist doesn't know about the name of the medications. So it is nothing but we can provide the explain well, it is a parcel of the good medication, and that definitely will be better for the patient, their health and their health. Next.
Thank you. 1 second, sir.
I would like to thanks to both.
Yeah. Okay.
Okay, if you miss, then don't worry. Any questions? That is slide number is that. Thank you for your attentions, and if it is that is okay. Okay. Any questions? Please raise your hands. And next now. Okay. I thanks to IPCO, Indu Kaka, IPCO Wala College of Pharmacy and Gujarat State Pharmacy Council, and my dear friend and team of the Montu Patel to provide me an opportunity to share my views with the dear pharmacists. Again, I really appreciate your help during the corona. You are really a good social worker of the society. Pharmacists, and one topic I should not be missed, the pharmacist, you can improve, or you can improve the image of the pharmacists in the society. We know the impression of the doctors, we know the impression of the nurses, but somehow the pharmacists have not too much front image of in the society. But in Corona, we have now somehow we are popular as a pharmacist, and hope so. The Corona vaccine should be invented by the pharmacist only. So good luck to all the pharmacist fellow, and thank you so much, Arsab and special thank yous to you. And after a long time, I have been seen in uh, as your picture. So any question? Maybe you can ask the question. Otherwise, we can have the next session.
Sir, there are a few questions from the
Yes,
audience.
please.
Yeah. The first question is, are the anticoagulants required in the treatment of corona or with the medication like antibiotic or antivirals?
And regulation for antibiotic and antiviral are there, but for corona, till there is no medication, so it is very difficult to say what the regulations should be need. But once it's come, that will be comes with the regulation only.
Yeah. Also, sir, thank you. Thank you so much for your words of wisdom and sharing your reflective knowledge with us that how the registered pharmacist can do better counseling of the patient for the betterment of the India. Thank you, sir. Thank you. Okay. Uh, the the aim of the education is the knowledge, not of the fact, but of the values. We are having with us the second speaker, Mr. Devang Tandil, Assistant Professor, Anand Pharmacy College, Anand, and he would be delivering the lecture on viability of drugs. Um, we will start with the session. Wait for it.
The times are changing, and so is the world around you. At CBM University, we help you prepare for the future, like we have with more than 10 lakh students. Come, join the educational legacy of 75 years. CBM University, enlightening the future generations.
Okay, I would like to request Dr. Veena Patel, Associate Professor and Head of Pharmacology Department, ISCP, Pharmacognosy Department, ISCP, to introduce Mr. Day1 Tandel, sir. Over to you, ma'am.
Thank you, Heta ma'am. Good afternoon to all. It is my privilege to introduce our next resource person, Mr. Day1 Tandel. He is presently working as an assistant professor in pharmaceutical chemistry department at Anand Pharmacy College, Anand. He has more than 7 years of research and teaching experience. His core areas of research are development and validation of bioanalytical method and dissolution enhancement of drug. He published 6 research articles in referred pharmaceutical journals. He presented 10 poster presentation in international, national, and state level conferences. He has also guided 4 PG students for their MPharm dissertations. Various awards and prizes are to his credit. Name a few. National level Pharma Innova Award 2014 for best student. Sir received first prize in poster presentation in one-day international conference on nanopharmaceutical therapeutics from laboratory to clinical practice in the year 2012. National level based research paper award 2016 in the field of pharmaceutics for the paper entitled improvement of dissolution rate of febuxostat using hybrid technique of spherical crystallization and solid dispersion during 54th annual day celebration organized by Indian Drug Manufacturer Association, Mumbai, and many more. With this brief introduction, I now invite Mr. Day1 Tandel to deliver a talk on viability of drugs. Over to you, sir.
Okay, thank you. Thank you very much, Dr. Veena Patel. Shall I start?
Yes, sir. You can start.
Okay. My voice is clearly audible?
Yes, sir. It is audible.
Okay. So I will start my screen sharing. My screen is visible to all of you?
Yes, sir. It is visible.
Okay. Okay. So a very good morning to or good afternoon to all of you. Myself, Day1 Tandel, Assistant Professor from Pharmaceutical Chemistry Department, Anand Pharmacy College. And today I would like to give you some basic informations about the bioavailability and bioequivalence study. So before we start, first of all, I would like to thank you very much to say about the Gujarat State Pharmacy Council and Indukaka Ipkola College of Pharmacy and the organizing principal Harsha madam, okay, and the coordinators Hita madam and Jinal madam and Usman sir for inviting me as a resource person for this 2 days online refresher course on the 26th and 27th September. So thank you very much. Thank you very much. So before we, without wasting a time, let us start with our session, that is bioavailability and bioequivalence study. So this is my today's route of presentation, that introduction of the bioavailability, types of bioavailability, objective of the bioavailability, measurement of the bioavailability, factors affecting bioavailability. Now next is bioequivalence studies, objective, types of the bioequivalence studies, bioequivalence study design, protocol, evaluations, and what are the regulatory requirements for the study and methods of enhancement of bioavailability of drugs. So let us start with the bioavailability. What is bioavailability? So according to the US Food and Drug Administration, the rate and extent to which the active ingredients or the molecule which is absorbed from the drug and becomes available at the site of action, that is called as bioavailability. Okay, so bioavailability is a simple thing that what we have understand that amount of drug that reaches to the site of action. Okay, so site of action means that maybe the organ, that may be the receptor. Okay, so that how much amount of drug that will be reached to the site of action, and that we can say it is called as bioavailability. And based on this, we can say that our formulation or our drug molecule, it is a therapeutic effective. Okay, so sometimes to check the therapeutic effectiveness, we have to perform this type of bioavailability study. So it is also defined as a rate and extent. Okay, so in how much amount or how much time a drug will be reached to the systemic circulation. So the rate and extent that which active drug is absorbed from the dosage form and becomes available in the systemic circulation. So why we are checking the systemic circulation? Because it is very, very difficult to check the amount of drug that reaches to the site of action, because we cannot quantify how much amount of drug that is reached to the receptor level. So okay, so so for that, we are calculating, or we are determining how much amount of drug that reaches to the systemic circulation, and we are assuming that if our drug is available in the systemic circulation, then it will definitely reach to the site of actions. So the relative amount of drug from the administered dose form which enters into the systemic circulation. Next is there are some of the route of administration that will affect the bioavailability. That is, first of all, influence of route of administration. Okay, that we all know that so many pharmaceutical dosage form that will be given by these four major routes, that is parenteral, oral, rectal, and topical. And we all know that the 100% bioavailability will be achieved with the help of parenteral route, that is IV or intravenous administration. Okay, and after this, the major route of drug of administration for most of the people, that is oral formulations. And the oral formulation, that is some barrier or there is some limitations for the bioavailability, because there are some problems related with the dissolution, disintegration, absorption. Okay, and that's why the bioavailability of the oral formulation, it is next to the parenteral. And after this, the rectal and topical route of administration, they have a very less bioavailability, or because they have some of the problems with the absorption also. As far as the concern about the subcutaneous injections as well as the intramuscular injections, they have a variation in the absorption or the bioavailability processes. Okay, so the amount of the drug that reaches to the systemic circulation, that is extent of the absorption, is called as systematic availability, or it is called as simply availability. So based on this, we have to calculate the bioavailable fraction. That means the amount of drug that we have administered and how much amount of drug that will be reached to the systemic circulation. Okay, that is a totally different thing. So what we have said, that amount of drug that is reached to the systemic circulation is called as bioavailable fraction. Okay, so how to calculate the bioavailable fraction? So there is a very simple formula, that we must have to know about the concentration of the drug that reaches to the systemic circulation divided by the administered dose, and we can easily calculate the fraction which is available for the site of action. So usually the bioavailability refers to the absorption of the drug from the gastrointestinal tract following by the oral administration of the dosage form. Okay, why we are more focusing on the oral dosage form? Because the oral formulation having its own advantage over the some another formulations like intravenous or parenteral formulation. For intravenous and parenteral formulation, your drug should be soluble in a different solutions. Okay, there are some stability issues with the intravenous administration. It requires one person to inject to the patient. Okay, so there is no patient compliance. Okay, stability issues. So that's why the oral formulations. Okay, so in the pharma market, oral formulations having a large market. Okay, or a huge market or a huge revenue. Okay, so we have to more focus on our oral formulation, how we can improve our oral formulations. Okay, so there are a lot of type of oral formulation available, that is solution, suspension, tablet, capsules, powders, and elixirs. So types of bioavailability. There are two types of bioavailability: absolute bioavailability and relative bioavailability. What do you mean by absolute bioavailability? So here we are comparing our formulation with intravenous formulation. Why? Because the intravenous formulation or IV formulation, that means it is completely bioavailable. Okay, that is 100% it is available at the site of action. So comparing with the IV formulation. So that absolute bioavailability is the drug that is fraction of administered dose which actually reaches the systemic circulation and ranges from 0 to 1. 0 means there is no absorption and 1 means that is a complete absorption. So here what we have to do, since total amount of drug that reaches the systemic circulation directly proportional to the area under the plasma drug concentration versus time profile. So here what we are going to do, we have to check our plasma concentration versus time profile of our oral formulation with IV. Okay, so IV માં કેવું હશે તો મને તમને દેખાય છે on my presentation on the right hand side, the upper part, the red color જે છે બરાબર છે graph, that is the for the IV formulation, and the blue color indicate that is our oral formulations. Okay, and that we have to compare. So the IV formulation having a 100% bioavailability, but the oral formulation there is a one disintegration, dissolution, absorption, and after this they will reach the Cmax, that is a maximum concentration in the drugs, and later on that will be excreted from the body. What do you mean by relative bioavailability? Relative bioavailability, that means we have to check our formulations bioavailability with some of the already available in the markets. Okay, that is an innovator company, okay, or some of the multinational company, and one domestic company is providing or manufacturing some of the formulation, then we have to compare. Okay, so that is called as relative bioavailability. So the fraction of the dose that is systematically available as an oral drug product. Okay, so here we are going to compare. Okay, so we are comparing with our standard formulation, not we can say standard, but we are comparing with our marketed already existed formulations. That is called as relative bioavailability. So there is a one simple formula to determine the absolute bioavailability, that is FA is determined by comparing the respective area under the curve and test product. Okay, and that we have to compare with the IV, because the IV having a maximum area under the curve. What is area under the curve? Area under the curve means rate or extent of the absorption of the drug from the formulations. Okay, so જેનો AUC વધારે હશે બરાબર છે, તો એની જે ફાર્માકોલોજીકલ એક્શન હશે બરાબર છે, એનું જે અમાઉન્ટ હશે એ સિસ્ટમિક સર્ક્યુલેશનમાં સૌથી વધારે હશે. Okay, so that is called as absolute bioavailability. And the relative bioavailability, now we have to compare with our reference or a standard or a marketing formulations. That is called as relative bioavailability. Now what is the objective of this bioavailability study? It's very simple, because without performing any bioavailability study we cannot sell our formulation to the market. So it's a primary stage of the development of the dosage form for the new drug entity and find its therapeutic utility. Okay, કયા formulations થી આપણને વધારે therapeutic activity મળશે એ જો check કરવું હોય તો then we have to perform the bioavailability study. Determination of the some of the influence of the excipient. Okay, because we are not giving our API, active pharmaceutical ingredient itself બરાબર direct ને direct આપણે આપતા નથી. Okay, there are so many excipients in the even injection, solution, suspension, and tablet and capsule. So we must have to check, is there any interaction or is there any influence of the excipient which is present in our formulation. And development of the new formulation. Obviously, if there is a problem with the existing drugs or existing formulation, then we must have to develop some of the new formulation, and we have to check whether our formulation it is better compared to the existing formulation or not. Okay, and sometimes there is a problem related like the processing factors, storage factors, stability, absorption, and that's why we have to develop the new formulations. Okay, day by day. So the company what they are doing, they are always developing some of the new formulations. Okay, for the betterment of the process. Okay, stability point of view, storage point of view, as well as the absorption point of view. Okay, and then simply that is comparison of the drug in a different dosage form or same dosage form with a different manufacturers. Okay, so there are so many more than 1,000 pharma companies are there. Okay, 1,000 manufacturing companies are there, and they are just comparing with one another, and they are saying that yes, my formulation is better than this, my formulation is exactly behaving like this. Okay, now how to measure the bioavailability? Measurement of the bioavailability with the help of two methods. One is a pharmacokinetic method and one is a pharmacodynamic method. For this pharmacokinetic method, there are two simple techniques, that is to prepare or to conduct the plasma level time study to do one another study, that is urinary excretion study. For a pharmacodynamic method, two another techniques are that is to check the acute pharmacological response, and the second one, that is a therapeutic response. Let me explain you in brief. That is, first of all, that is blood analysis. Blood analysis is very simple. That what we have to do, we have to administer one simple or single dose of our drug to the patient, and we have to collect the blood at a different fixed time interval. Okay, and we have to check how much amount of drug it is present in the blood sample, and we have to prepare one graph, that is a plasma concentration versus time profile. So based on this plasma concentration versus time profile, we will get some of the information of our dosage form. That is a major three information that is very important. That is peak plasma drug concentration, time of the peak plasma concentration, and area under curve. What is Cmax? Cmax is a maximum concentration of drug, okay, that reaches to the systemic circulation. That is called as Cmax. What is Tmax? A time required to achieve the peak concentration of the drug that is administered. It is called as Tmax. So that will give the extent of rate of absorption. Okay, so with the rate of absorption, we have to calculate this Tmax. Okay, now what is area under the curve? Area under the curve, that is very important because it's measurement of the extent of the drug bioavailability, or we can say extent of the drug absorption. Okay, starting from the absorption point of view and at the end of the excretion. આખો area આપણને બતાવે છે that how much amount of drug that will be remain in the systemic circulation from this to this time period. So next is urinary excretion data. Now, why we are going to do this type of study, that is urinary excretion? That means obviously whatever we are eating, whatever we are taking into our body, that will be excreted from our body. So we have to check whether how much amount of unchanged form of drug that will be excreted from our body. Okay, so for that study, your drug, it should be more than 20%. It should be excreted more than 20% in the unchanged form. And then we can go for this type of urinary excretion study. And what we are concluding here, that is a cumulative amount of drug that is excreted in the urine is directly proportional to the extent of systemic drug absorption. Okay, એટલે શું કહે છે કે જેટલું વધારે યુરિનમાં એક્સિડ થશે એનો મતલબ એ છે કે એનાથી વધારે એટલું જ systemic circulation માં absorb થયું છે એટલે બહાર નીકળશે. Okay, so for that urinary excretion study, there are three important parameters that we have to study. That is maximum urinary excretion rate, that is dxu by dt. That is nothing but obtained from the peak plot between the rate of urinary excretion data versus time. Okay, so at which time point we will get the maximum excretion of our drug. Time for the maximum excretion rate, that is a maximum time required to reach a maximum excretion rate. Okay, kaya time point પર સૌથી વધારે excrete થયું છે એ. અને બીજો point, cumulative amount of drug that is excreted from our body. So these are the pharmacokinetic methods. Next is pharmacodynamic method. Pharmacokinetic method, it is also called as indirect, because actually here what we are doing, we are just checking that how much amount of drug that is reaching the systemic circulation. Okay, now we have not performed any pharmacological factors or pharmacodynamic studies. So these are called as indirect method, and the direct method, that is a pharmacodynamic method. In this, what we have to do, we have to check these two methods or perform the two methods, that is acute pharmacological response. Acute means જેવું તમે ડોઝ આપશે એટલે તરત તમને એનો રિસ્પોન્સ જોવા મળશે, that is called as acute pharmacological response. So when the bioavailability measurement by the pharmacokinetic method is somewhat difficult, and we can go for the acute pharmacological effect methods. That is for that purpose, what we have to do, again we have to administer a very single dose to the patient, and we have to check, okay, that what will be the response. Okay, like અહીંયા તમને બતાવ્યું છે કે pupil diameter માં શું ચેન્જ થાય છે, okay, blood pressure માં શું ફરક પડે છે, electrocardiogram માં શું ફરક પડે છે. Okay, so that we have to check in this acute pharmacological response. And the second one method, that is a most reliable method, because this is a clinical response. Clinical response, that means our pharmacological response, that is our desired response. Apart from this, how many other effects we are monitoring, that is called as clinical response. Okay, and what we can say, that is a therapeutic response. So this is the best clinical response of the drug that is intended to be used in this method. Okay, એની અંદર ઘણા બધા models છે to check the anti-inflammatory activity, to check the anti-sclerotic activity. Okay, but ઘણા બધા models available છે, and based on this you can calculate or determine. Now just go for the briefly, that is factors affecting bioavailability. There are so many factors affecting the bioavailability, that is drug related factors, pharmacological factors, sometimes it is always pharmaceutical factor, that is dosage form related. So I will explain you a very brief or some of you, that is salt form. Salt form, that means nowadays most of the most of the our drug, okay, it is in a salt form. Why it is available in a salt form? Because the itself active pharmaceutical ingredient having problem regarding the dissolution. Okay, solubility hoti nothing. So we have to convert into this salt form, and we can say that yes, the salt form having a more solubility, okay, compared to the API itself. Okay, now the crystal form, there are so many polymorphic forms are available. Okay, so the polymorphic forms which is available having a different solubility and different dissolutions. Okay, so based on this we can give some predict the bioavailability. Now next is particle size. Obviously that particle size, what we have to know, as the particle size decreases, the dissolution or solubility of the drug is increases. Okay, now the degree of ionization, how the degree of ionization that will affect. So for that you must have to know about the Henderson-Hasselbalch equation, and we must have to know about the ionization process of our drug molecule into the stomach as well as in the intestine. If our drug is a weak acidic in nature, then absorption and ionization will be very good from the stomach. So that we can calculate from based on this a pK value. Okay, so if the pK value is somewhat basic side, then the absorption from the small intestine is very good. Okay, so this type of information, okay, that we will get from the degree of ionization. Now what are the pharmacological factors that will affect? That is a pH. Obviously that the starting from the esophagus, okay, stomach, intestine, small intestine, large intestine, the pH is varying. Okay, so we must have to consider that. That is very important, because pH will affect the ionization of the process. Okay, some another parameter, that is first pass metabolism. Yes, first pass metabolism, that is a major problem with the oral formulation, that whatever the absorption from the GI route, that will first go to the liver and then it will be transferred into the tissue distribution. Okay, so that will problem with the oral formulation. And some of there are drug-drug interaction. Obviously if you are treating the patient with so many disease, then drug-drug interaction are there. Now what is bioequivalence? Bioequivalence means a drug from a two or more similar dosage form reaches to the general circulation with a relative rate and extent, then dosage forms are termed as bioequivalent. Means both the formulations having a similar, not the identical, what we are saying, that similar rate and extent of absorption. Okay, જો જોવા મળશે then આપણે કહીશું bioequivalent. Okay, તો તમે રાઈટ હેન્ડ સાઈડ ઉપર મેં જે ગ્રાફ બતાવ્યો છે તમે કહી શકો કે યસ, the red one, that is an innovator company. Okay, and the blue one, that is our generic company. Okay, so innovator, most of these nowadays the most of the pharma companies, they are just waiting that at which time the innovator company's patent will be expire and we will launch our product. So for that purpose they have to show that this type of bioequivalent studies or bioequivalent data. Okay, and based on these they can claim, okay, they can file the patent in the IPR. Okay, another term is that is a statistical significant differences are observed in the bioavailability of two or more formulations, that we can say it is called as bioinequivalence. Okay, તમે જોઈ શકો છો કે ગ્રાફમાં દેખાય છે કે ઓકે બ્લુ કલર જે છે that is somewhat different and red is okay different, same formulation, same API. Okay, 150 મિલીગ્રામ બુપ્રોપિયોન બતાવ્યું છે. ઓકે, બ્રાન્ડ નેમ અલગ છે, મેન્યુફેક્ચરિંગ અલગ છે. તો આપણને જોવા મળશે કે જે પ્લાઝમા કોન્સન્ટ્રેશન વર્સિસ ટાઈમ પ્રોફાઇલ જે બંનેનો અલગ અલગ છે, તો એને આપણે બાયો ઇક્વિવેલન્સ કહી શકીએ અને એને કહેવાય બાયો ઇનઇક્વિવેલન્સ. What is the objective of this bio inequivalence, okay, or bio equivalence study? Okay, obviously there is a significant changes are made in the manufacture of the market formulation, because yes, each and every pharmaceutical company, they wanted to sell their products. Okay, so there are so many pharmaceutical companies are there, competition is very very high. So how we can say that our pharmaceutical product is exactly matched to the innovator company? So for that purpose we have to perform this type of bioequivalence study. Okay, so that is bioequivalence study, that is nothing but a comparison, okay, comparison with our formulation with our innovator companies. Now these are some of the basic terminologies it is used to understand the bioequivalence study, that is chemical equivalence, pharmaceutical equivalence, and therapeutic equivalence. What is chemical equivalence? The term indicates that two or more drug products contain a labeled chemical substance as an active ingredient in the same amount. That means a two formulation is there, okay, having a same active pharmaceutical ingredient with a same dose, but the use of excipient, okay, it is totally different. So that we can say અને જો same study આપશે તો આપણે કહીશું that is a chemical equivalence. Now in the pharmaceutical equivalence, and કોને કહેવાય? Drug product that contains identical amount of the active pharmaceutical ingredient, like a same salt, same ester, okay, same therapeutic moiety, but here not necessarily containing same active ingredient, same inactive ingredient. બરાબર, તેને કહીશું આપણે pharmaceutical equivalence. Now the therapeutic equivalence. In this therapeutic equivalence, these are the drug products that are coexisted with the therapeutic equivalence only if they are pharmaceutical equivalent and they can be exposed to have a same clinical effect. બરાબર, same therapeutic effect તમને જોવા મળતી હોય તો તમે કહી શકો છો that is called as therapeutic equivalence. Now types of bioequivalence study. So types, there are two ways to perform this bioequivalence study. One is a in vivo and second one is a in vitro. In vivo, that is we all know that is oral or oral formulations we have to give, okay, oral immediate release product we have to give, and we have to check that how much amount of drug that reaches to the systemic circulation, okay, and it is always performed for the so many dosage form, because immediate release form is available, sustained release formulation is available, okay. So at that time we have to perform this type of in vivo study. What is in vitro study? Now in vitro study we are performing when one pharmaceutical company is there, okay, suppose he is manufacturing one tablet formulation having a propranolol, okay, like a 50 milligram. Now the same pharmaceutical company wants to introduce or wants to sell, okay, one another formulations having a same, okay, same excipient, but there is a slight change in the dose of the drug, okay, 50 ની જગ્યાએ લોકો કરે છે કે 30 milligram. So at that time again we have to perform this bioequivalence study or not. So the regulatory companies, okay, US FDA છે, બીજા બધા ઘણા બધી regulatory company છે, they have said that you are exempted to perform this type of bioequivalence study, okay, for this type of conditions. બરાબર, તો એ ટાઈમ પર બીજા બધા જે conditions છે like a pharmacokinetic profile, qualitative composition, API, excipient, આ બધી વસ્તુ સેમ હશે તો તમને બાયો વેવર મળશે, okay. So at that time just you have to perform the in vitro dissolution profile. There is no need to perform the bioequivalence study or slightly reformed or manufacturing methods. બરાબર, slightly તમે change કરો છો process માં, okay, તો તમારે આ વસ્તુ કરવાની રહેશે. Now what is the experimental study design for the bioequivalence study? Now for the design and evaluation of the well controlled bioequivalence study, it requires inputs from the pharmacokinetic study operators, okay, બધાથી એને support જોઈશે. Obviously that is a teamwork, pharmacokinetic study operators, statisticians, clinicians, bioanalytical method development, and others. Basic design for the bioequivalence study is determined by the scientific answers. બરાબર, તો એના માટે આખું એક પ્રોટોકોલ જેવું છે, બરાબર, અને સ્ટડી કઈ રીતે થાય છે તેના ચારમાં ડિવાઇડ કરેલી છે, okay. First one, that is completely randomized, second is randomized block design, third one is crossover design, and the fourth one is Latin square. Now the first one, that is completely randomized, Now in this completely randomized, all the treatments are randomly allocated among all the experiment subjects. બરાબર, સપોઝ કે 20 નું ગ્રુપ બનાવ્યું છે, બરાબર, એ 20 માં 1 by 1 બધા ડોઝેજ ફોર્મ આપતા જશે, એક્સપેરિમેન્ટ પરફોર્મ કરતા જશે, અને પછી એમાંથી રેન્ડમલી લોકો ટેસ્ટિંગ માટે લેશે. એને કહેવાય છે કમ્પ્લીટલી randomized design. So the randomly selected non-repeating numbers from among these label for the first treatment and repeat all for other treatment. That is completely randomized design. Now second is randomized block design. Okay, now what is the difference between these two, completely randomized and randomized block? Completely randomized માં બધા મિક્સ હશે, like a man, female, okay, પછી age, પછી એની weight કેવું છે, બરાબર, એનો બોડી માસ ઇન્ડેક્સ, આ બધા પેરામીટર જે છે બધું મિક્સ હશે, એને કહેવાય completely randomized. While in the randomized block design, what we are going to do, we are going to prepare one group, okay, the group having the same homogeneous characteristics, like આ એજ નો એક ગ્રુપ બનાવ્યું છે, આ એજ વાળાનો એક ગ્રુપ બનાવ્યું છે, પછી આ વેટ વાળાનો એક ગ્રુપ બનાવ્યું છે, એના જેવું આપણે જો પછી ટ્રીટમેન્ટ આપશું તેને કહેવાય છે randomized block design. Okay, now what is a crossover design? Crossover design, that means આ એક ગ્રુપ છે, એને પહેલા પ્રોપ્રાનોલોલ 50 મિલીગ્રામ થી ટ્રીટમેન્ટ આપીને પછી ચેક કર્યું. ઓકે, આફ્ટર ધ વોચિંગ પીરિયડ, ઓકે, કોઈ બીજી જ ફોર્મ્યુલેશન થી બીજા ડ્રગ થઈને આપણે ચેક કરીશું, પછી ફરીથી થર્ડ ટાઈમ જ્યારે આવશે ત્યારે ફરીથી પ્રોપ્રાનોલોલ ચેક કરીશું, એને કહેવાય છે that is called as crossover design. ઓકે, તો આપણે ક્રોસ ચેક કરીશું અને દરેક સબ્જેક્ટ છે, દરેક હ્યુમન વોલન્ટિયર કે પેશન્ટને આપણે બધી જ ટ્રીટમેન્ટ આપીશું અને ચેક કરશું કે is there any effect or not. બરાબર, આપણે સ્ટેટિસ્ટિક્સ માં કંઈક અલગથી રિઝલ્ટ જોવા મળે છે કે નહીં. Fourth one, that is a Latin square design. Now the Latin square design, that is nothing but they have divided the human volunteers and treatment in one Latin square design. અને એની અંદર જસ્ટ બીજું ચેક શું છે, comparison. Okay, the design is useful to compare the earlier one when these 3 or more treatments are to be compared. Now this is a study protocol. Okay, so bioequivalence study protocol, we must have to go through this step-by-step process, and we have to must consider about this. That is a concern about the principal investigator, project number, date, study objective, study design, which will be the test product will be there, which the reference product will be there. Okay, કયો ડોઝ હશે, સેમ્પલ કલેક્શન એટલે કે એનું જે શેડ્યુલ છે આપણે કઈ રીતે કરશું, પેશન્ટ ઘરે હશે કે પછી આપણે હોસ્પિટલમાં હશે, એને ફાસ્ટિંગ કેટલું રાખવાનું છે, 10 કલાક પહેલા રાખવાનું છે કે 5 કલાક પહેલા રાખવાનું છે, which analytical method we are going to perform for the determination of the concentration of drug. Subject selection, okay, કે ભાઈ કયા કયા એરિયામાંથી આવે છે, બરાબર, એની મેડિકલ હિસ્ટ્રી શું છે, એનું ફિઝિકલ એક્ટિવિટી શું છે, ઓકે, એનું લેબોરેટરી ટેસ્ટ, okay, which are the some of the inclusion and exclusion criteria, okay, what will be the clinical procedure, okay, for the taking the samples and further performing any test. બરાબર, કયા કયા બાયોલોજિકલ સેમ્પલ્સ તમે કલેક્ટ કરશો, યુરિન કલેક્ટ કરશો, પ્લાઝમા કલેક્ટ કરશો કે બ્લડ કલેક્ટ કરશો, ઓકે, ઇથિકલ કન્સિડરેશન્સ લીધેલું છે કે નહીં, હ્યુમન વોલન્ટિયરનું પોતાનું કન્સર્ન છે કે નહીં, ઓકે, આ બધી વસ્તુઓ જે છે that is included in the bioequivalence study protocol. Now next is evaluation of this bioequivalence study. After performing the bioequivalence study, there are so many statistical tools that we can go for the calculation of the bioequivalence study, that is the analytical methods. Obviously nowadays, because of the available of so many sophisticated instruments like LC-MS, MS, TMS, we can determine the even a picogram level of the drug in our biological fluid. So we have to go for that type of analytical method because they are very accurate, precise, and very specific. Okay, and some of the pharmacokinetic evaluations that we have already discussed, that is Cmax, Tmax, area under the curve. Okay, now why we are applying the statistical tools to analyze our data, because these are, we are dealing with the human volunteers, we are dealing with the pharmacokinetics and dynamic study. So in this type of study, so many errors are there. That may be a human error, that may be a practical error. So to nullify or to nullify this error, we are applying some of the statistical tool and we will get some of the fruitful results. Okay, that's why we are applying this ANOVA, one-way ANOVA. Okay, there are so many statistical tools that you can apply. Now these are the regulatory requirements for the bioequivalence study. I'm not going take into the detail, but just as a explaining part. Okay, so I have taken from the one of the review article, and we have compared the so many criteria with our 6 ગાઈડલાઈન છે. ધેટ ઇઝ US FDA છે, યુરોપિયન ગાઈડલાઈન છે, કેનેડિયન ગાઈડલાઈન છે, CDSCO ઓકે ઇન્ડિયન ગાઈડલાઈન છે, પછી છે સાઉથ આફ્રિકા અને સાઉથ ઈસ્ટ એશિયન નેશન. ઓકે તો આ 6 કન્ટ્રીનું કમ્પેરિઝન જસ્ટ બતાવ્યું છે તમને. જનરલ બતાવ્યું છે. જનરલમાં શું છે સિંગલ ડોઝ ઓકે જે સ્ટડી કરવામાં આવે છે બરાબર તો મલ્ટિપલ ડોઝ બહુ નથી કરવામાં આવતી. શું કરવામાં આવે છે ધેટ ઇઝ અ સિંગલ ડોઝ નોન રેપ્લિકેટ ક્રોસ ઓવર ડિઝાઇન. ઓકે ધેટ ઇઝ preferable for all the countries. Second, that is a long half-life drugs or highly variable drugs. એના માટે શું કરે છે લોકો કે parallel design. Okay, they are conducting a parallel design for this type of drugs. Okay, and that is for all the countries. What is a blinding? બરાબર blinding means પેશન્ટને નથી ખબર હોતી કે પછી ડોક્ટરને ખબર નથી હોતી. તેમાં શું આવે છે કે it is not specified for so many countries but it is specified for the Canadian country. That is double blind study we are conducting. Fourth one, that is a number of subjects, and how many subjects, minimum how many subjects we have to take. So as far as you look at this slide, that is not less than 12. Okay, more than 12 હોઈ શકે છે but not less than 12. But for the CDSCO, that is our Indian guideline, that is not less than 16. બરાબર બાકી બધા માટે થોડું સેમ છે. Now the gender of the subjects. Okay, male લેવાનું કે female લેવાનું તો બધામાં obviously common, that is male and female, you have to consider both. 6, that is a female subject. While selecting the female subject, you must have to take some of the precautions. Why? Because if the female is having a pregnancy or taking a contraceptives or taking some another medication, then we cannot include in our study. Okay, so for that CDSCO has given some of the guidelines that we have to ignore the patients having a pregnancy or having a taking and contraceptives. We have not to include it in our study. 7, that is a replacement of the subjects on the withdrawal or dropouts. Yes, if after giving a dose or after giving drug treatment, if the patient found some of the adverse effect or side effect, then what we have to do? Okay, so for that they have given the guideline that we have to remove from the study and not include it for the further study. બરાબર એમને તરત કાઢી નાખવાનું છે પણ કરવાનું નથી. ઓકે ફરીથી what is age? Obviously more than 80. Okay, બધા healthy volunteers આપણે always લઈશું. Body mass index, obviously that is 18.5 to 30 kg per meter square. It is most for all the countries. Next strength of the dosage form. Yes, strength means how much amount of dose. Okay, the dose કેવો હોવો જોઈએ તો obviously from the highest, okay, highest strength થી તમે ચાલુ કરો એ વધારે preferable રહેશે. 11, that is a single or multiple dose. So in these most of the country they are following the single dose study, except from these European countries that they are following the multiple dose studies, acceptable only in case where it is not possible to carry out the single dose studies. Okay, next is genetic phenotyping. Okay, genetic phenotyping means genetic change હોય એકદમ અલગ રીતનું હોય બરાબર તો એ કન્સિડર કરી લેવાનું બરાબર, and it is not specified in some of the country like Canadian or USA. Next is the parent drug or the metabolite. Okay, obviously yes, sometimes our drug that easily metabolize. So what we are going to do, we are taking the unchanged form of the drugs, we are determining the metabolites or what. So here લખ્યું છે કે parent drug analysis is considered the most of all the cases except for certain case and the parent drug level are too low to allow the reliable analytical measurement. બરાબર તો parent drug જ કરવાનું છે અને metabolite કેવું કશું જ estimate કરવાનું નથી. Okay, now next is emesis and vomiting. Yes, and most of the time if the human volunteer or patient will take the medicine, they are vomiting or nausea will be there. તો એવું થાય તો એ પેશન્ટને આપણે એમાંથી સ્ટડીમાંથી કાઢી દઈશું. ફાસ્ટિંગ કેટલું હોવું જોઈએ? તો તમે જોશો બધામાં ઓલરેડી સેમ છે, 10 hours before and after the drug administration, that is 4 hours. So during these 14 hours તમારે કશું જ ખાવાનું નથી. These are the guidelines. Now the food specifications. Okay, which type of food okay you have to take after these medications or before the medications. Okay, high fat ને આ બધું જે છે થોડું અવોઇડ કરવાનું during this type of study. Fluid water intake બરાબર જે પણ નોર્મલ લેતા હોઈએ છે એ જ લેવાનું છે. સેમ્પલિંગ જે ટાઈમ છે that is 12 to 18 samples બરાબર 12 to 18 samples જે છે ઓકે એ જ રીતે તમારે લેવાનું છે. Wash out period, obviously If the same human volunteer go for the some another study, then there is a washout period. बराबर, completely drug तुम्हारी बाहर निकली जाएगी चाहिए. And these are the some of the parameters that you have to calculate. Now, move toward the last topic from my presentation, that is methods for the enhancement of bioavailability study. Okay, now why we have to enhance our bioavailability study? Because nowadays, whatever the innovator company, whatever the innovations is there, okay, medicines तो बनी જાય છે, बहुत सारी medicine बने છે, okay, pharmacological activity પણ बहुत सारी છે, okay, but this drug having a serious problem, that is low bioavailability. Okay, so amount of drug, okay, desired amount of drug which is reach to the site of action is not possible. Okay, why this? So I have listed out some of the problem, or we can say causes of the low bioavailability, that is first pass effect. બરાબર, જેટલું પણ આપે છે બધું ફર્સ્ટ પાસ માં એટલે કે લીવર અને મેટાબોલિઝમ કરી નાખે છે અને પછી જે બચે છે બરાબર તમે સાઇટ ઓફ એક્શન સુધી પહોંચશે. સેકન્ડ વન, that is a major problem, that is poorly water soluble and slowly poorly or oral drugs. બરાબર, ડિસોલ્યુશન મીડિયામાં એટલે કે 0.1 મોલર HCl છે, pH બફર છે, એમાં જલ્દીથી એ ડિસોલ્વ નથી થતું. ઓકે, so there is some limitations. બીજું છે એજ છે, સ્ટ્રેસ છે, ડિસઓર્ડર છે, બરાબર, these are also affecting the low viability. Okay, sometimes chemical reactions will be there, okay, between drug food interaction, drug interactions, okay, and metabolism by the luminal microphone. Now, before we go for the methods for the enhancement of bioavailability, first we have to understand this is a very basic, that is biopharmaceutical classification of the drugs. Okay, so four major classes છે, solubility high and permeability high, that is absorbed well, there is no problem related with this class of the drug. Class 2, that is solubility is very very low, but the permeability is high. So the absorption is variable, because for the absorption, your drug, it should be in a soluble form, not in an ionized form, but in a soluble form. That is very very important. Okay, the example is nifedipine and carbamazepine. So the challenges for this type of drug is formulation. તમારે formulation એવી રીતે design કરવાનું છે કે એનું resolution થોડું improve થાય. Third one, that is solubility સારી છે, but the permeability is low. Again, the absorption is variable because the permeation is a limited factors like insulin and metformin. Okay, so approaches that we are going to use some of the permeation enhancer to improve what formulation. In the class 4, there is a major limitations, that is a low solubility and low permeability. Okay, so both the limitations છે, okay, for the class 4 drugs. Now there are some of the methods to enhancement of the bioavailability for the enhancement of dissolution rate. Okay, so dissolution rate તમારે જો enhance કરવું હોય, dissolution process તમારે જો enhance કરવી હોય, so there are so many techniques. Okay, બહુ બધી techniques છે, મેં થોડીક જ technique અહીંયા થોડું તમને બતાવ્યું છે. First one, that is micronization and nanonization. Micronization means you have to decrease the particle size of our drug molecule, so that will be dissolved well and absorbed well. Nanosization, that means this is the nanoparticles which is available in the market, and this is booming in the pharmaceutical companies. But because of some of the stability issues of our formulation, nanoparticle is not as like our foreign countries. બરાબર, nanoparticle એટલું બધું ઇન્ડિયામાં એટલું સક્સેસફુલ નથી, તો આમ છતાં બી છે ઘણા બધા formulations. બરાબર, use of the surfactant, obviously we are going to use some of the surfactant in our suspension, emulsion. Okay, so that will give you a better dissolution process. Okay, so use of the salt form, that is obviously, and for this converting the salt form, it requires some of the chemical functional groups that we can convert into the salt form. Alteration in the pH of the micro environment. Okay, એના માટે તમારે શું કરવું પડશે, તમે નામ સાંભળ્યું છે ઓરો ડિસ્પર્સિબલ ટેબ્લેટ. ઓકે, પછી ફ્લોટિંગ ટેબ્લેટ્સ. ઓકે, તો આ બધી વસ્તુઓ જે છે એની અંદર આવશે કે ભાઈ pH થોડી અલ્ટર કરશું તો એનું એબ્સોર્પ્શન જે છે, ડિસોલ્યુશન પ્રોસેસ છે, that will be changed. Now the amorphous and the solvents, as we all know that the crystalline structure and the amorphous structure, the stability issues is there, very very less stable, but the solubility and dissolution is very very high. And as far as compared with the crystal structure is stable enough, but there is a dissolution limitations. Okay, so these are some of the ways that we have convert our crystalline structure into some of the amorphous form, and we can improve the dissolution profile. This is the one another technique that is solid dispersion. Solid dispersion, that means we have to use some of another excipients like a polymer, like a beta cyclodextrin, PG4000, PG6000. These are the polymers. These are hydrophilic polymers which will enhance the solubility of our drug molecule by the inclusion complex formation. Whatever are the techniques they improve કરી શકે છે. And the last one, that is molecular encapsulation. Molecular encapsulation, that is nothing but a large polymer structure is there and we are incorporating our drug molecule into this polymer structures. Okay, so that really enhances the dissolution process. Another approach, that is enhancement of drug permeability. પેલું હતું dissolution, ત્યારે અહીંયા આવશે permeability. Okay, so here there are five techniques are there. First one, that is a SMATS and SNATS. What is SMATS? Self micro emulsifying drug delivery system. Okay, self micro emulsifying मतलब કે એ હોય પાવડર ફોર્મમાં, જેવું તમે સ્ટમકમાં ઓરલ થ્રુ લેશો, બરાબર છે, તો automatically that will be dissolved or form a emulsion. એને કહેવાય છે self micro emulsifying drug delivery system. અને એનાથી એક થોડું એડવાન્સ, that is self nano emulsifying drug delivery system. Okay, so for preparing this type of formulation, you require some another excipients, that is a surfactant, co-surfactant, some of the oils, okay, to dissolve this type of drug molecules. Next is solid lipid nanoparticles. In the solid lipid nanoparticles, what we are going to do, we are taking this one type of polymer, that is PEG. બરાબર, તમને ડાયાગ્રામમાં થોડું બતાવ્યું છે. ફોસ્ફોલિપિડ્સ છે, drug molecule છે. So the drug molecules that we will incorporate it into this core molecule, barabar, and surrounded by the phospholipid monolayer. So this type of preparations are also there, that is solid lipid nanoparticles. Penetration enhancers. Okay, some of the penetration enhancers are also available to enhance the permeability of the drug molecule. And the last one, that is the liposome. Okay, the liposome, again the same thing, that is a one core molecule જ્યાં આપણે આપણો drug molecule મૂકશું, surrounded by the phospholipids. Okay, so this is the way that we can transfer our drug molecule, okay, drug to the site of action. Last one, that is enhancement of drug stability. Okay, so many formulations or so many drugs having stability issues. So how we can improve that? By these three techniques, that is complexation. બરાબર, તમને દેખાય છે કે drug, we have to incorporate the cyclodextrin, inclusion complex will be there, so the stability will be improved. So there is a very less chemical reactions between our drug and the enzyme. And the use of metabolism inhibitors, or what we can say, some of the drugs are always suggesting, doctors suggesting that by આ drug આની જોડે નહીં લેવાની, કે આ drug આની જોડે લેવાની. કેમ? Because some of the drugs, they are called as enzyme inducers. બરાબર, સાઇટોક્રોમ જે P450 જે છે એનું કોન્સન્ટ્રેશન વધારે છે, બીજી ડ્રગ મેટાબોલિઝમ થઈ જાય છે. આના માટે આપણે આ યુઝ કરી શકીએ છીએ. And the last one, that is entry coated, that is again a pharmaceutics part, is very very important for this development of the dosage form, that is entry coated. Okay, we have to coat our drug molecule and administer it into the stomach or intestine, so they will release the drug based on our pH and sometimes based on our environment of the stomach or intestine. So, okay, so these are the how we can improve the drug stability. And lastly, thank you very much for giving me opportunity to express my view. So thank you very much once again.
Thank you so much for accepting our invitation and for sharing the very informative views about the topics. Sir, we have some questions.
Yes.
First is, what is 90% confidence interval means in interpretation of B study?
Yes, in this 90% confidence interval, there is one graph. તો એ કહેવામાં આવે છે કે ગ્રાફની આજુબાજુ 90% જે છે, that means 90% માં કેટલા ફોર્મ્યુલેશન, બહુ બધા સબ્જેક્ટ્સ હોય છે, બરાબર, બહુ બધા સબ્જેક્ટ્સને આપણે એક ટ્રીટમેન્ટ ચાલુ કરી હોય છે, તો કેટલા સબ્જેક્ટ જે છે, બરાબર છે, એ 90% ના ઇન્ટરવલ્સમાં આપણને મળે છે. અને 10% એટલે કે જે એકદમ જ અલગ રીતનું રિઝલ્ટ તમને આપશે, બરાબર. તો એના માટે આ કોન્ફિડન્સ ઇન્ટરવલ જે સ્ટેટિસ્ટિકલ ટૂલ છે એ બરાબર એ યુઝ થાય છે ટુ ચેક ધી ખરાબ રિઝલ્ટ આવે એને બહાર દેખાશે અને સારું રિઝલ્ટ આવે એ આપણને એ એરિયામાં તમને દેખાશે.
Okay, one more is there, sir. From the different methods, which is the best and effective method for to determine the viability of drug?
according to my point of view, the pharmacodynamic method is actually more reliable, because in this method, actually, we are going to correlate our drug with our pharmacological activity or therapeutic effectiveness. Pharmacokinetics is an indirect method. Okay, so we are assuming that આટલું પહોંચે છે તો આટલી activity મળશે જ. So according to my point of view, the pharmacodynamic method is the most reliable method, because we are correlating our API or active pharmaceutical ingredient effect with our pharmacological effect. બરાબર, એ આપણે ચેક કરીએ છીએ.
Okay. Is the dicyanocobalamine a bioavailable form?
Dicyano?
Cobalamine.
No, actually, it's one of the type of vitamin, so it is not so much bioavailable. That's why the dose of this type of drug is very, very less.
Yeah. And one last is there, sir. Can we see the relative bioavailability as bioequivalence?
Yeah.
Okay. Thank you so much once again, sir.
Yes.
It is very nice to having with us you today. Thank you. Two sessions has been completed, dear participants. Now it's time for taking the lunch. So we are having a lunch break of half an hour, and we will start with the rest of two plenary sessions of today by 2 p.m. Okay, so we will meet on the same platform. Same link is on, so you can join the meeting by 2 p.m. by clicking on the same link.
Oh, you can also stay here.
And you can also stay on the meeting, no issue with that. Okay, thank you. Thank you, sir. Welcome back all. Moving towards the next plenary session, we are having with us Dr. Usman Gani K. Chalotia sir, Professor, Associate Professor and Head of Department of Pharmaceutical Chemistry and Analysis, ISCP. He would be delivering a lecture on rational use of drug. I would like to request Ms. Khushbu Chauhan, Assistant Professor ISCP, to introduce Dr. Usman Gani K. Chalotia sir. Over to you, ma'am. Thank you. Good afternoon to everyone present here. It is my honor and privilege to introduce my own sir, Dr. Usman Gani K. Chalotia sir. He is currently working as a head of the Department of Pharmaceutical Chemistry and Analysis at IICP New Vallabh Vidyanagar. He has earned his B.Pharm, M.Pharm and PhD degree from Sardar Patel University at Anand. He has a total of 11 years of experience in this period, and he has published one book and 66 scientific research papers in various national and international journals. He has guided 16 postgraduate students so far and was a head of 4 consultancy projects. He has been honored with PD Sethi Award for the development and validation of thin layer chromatography under the category of Certificate of Merit for 3 published research manuscripts. He is a recipient of several grants from Coast, GSBTM, and GSPC for organizing state and national level conferences. Recently, he has got recognition as PhD supervisor by Gujarat Technological University, Ahmedabad, as well as Sharthar Vidyamandal University at Anand. Today, he is going to deliver a topic on rational use of medicine. On behalf of IICP family, I welcome you, sir, to the refresher course. Thank you.
Thank you very much, Khushbu. And a nice way she delivered my profile. So thank you very much. Does it my voice is audible?
Yes, sir, you are audible.
Yeah. Thank you. So today I'm going to share a few things. But I'm not able to share the screen.
1 second, sir. Try now.
Yeah, thank you. Yeah. So first of all, I'm very thankful and congratulate to coordinator and co-coordinator of the today's 2 days event of the refresher course, which is sponsored by the Gujarat State Pharmacy Council and organized by the Indu Kakai College of Pharmacy. And I'm very thankful to both the coordinators and co-coordinators to invite me and give an opportunity to share a few things of with respect to the rational use of drugs. So very thank you to both of you. And now I would like to start the come on the topic that is rational use of the drug. Okay, ગુજરાતીમાં જેને કહેવાય આપણે કે દવાઓનો વ્યાજબી ઉપયોગ. So this pictures represent a very good thing. That is, see the tagline which is mentioned in the pictures that use medicines rationally. Okay, don't let yourself be flooded with them. Okay, so see some of the peoples I have seen even in my family, some of the members are also there, they are having a tendency or psychology, it's a psychology, they are always if anything is happened they are not bear it. Okay, suppose headache is occur or happen with them, so they immediately take a pill. Okay, so it is not the solution. Okay, because you all are aware that the NSAIDs are having a very bad side effects, and the we have to avoid as much as possible to take medicines. Okay, so now see the when we die not of the disease but from the medications. Not everyone is infected, but surely everyone is affected. Okay, so today our scenario, we are observing the COVID pandemic. In the situations, we observe that the see many of the peoples are very curious, even though they are not consulted with the doctors and they are taking azithromycin as HCQ medicines, which is anti-malarial, and all the pharmacists are aware with this. Okay, even though they are taking the doxolin 400 milligram tablet half on twice a day, half of tablet. It's a bronchodilator or COPD. It is widely used, that drug. So these are the things which is irrational, we can say. If you have the some symptoms, might be it is not because of the COVID-19. It is only because of the it's a simple flu or cold. Now, there are many factors which are influence the use of medicines. So here the treatment of the choice, see there are different categories with respect to the manufacturers which are influence the use of medicines. One is intrinsic, second one is societal, work group, workplace, and information. So in the intrinsic, that is prior knowledge and habits is required. Societal, the social, cultural, and factors which are very, very important, even though the economic and legal factors are also affected. Even the work group, the authority and supervisions and relationship with the peers, which is very, very important in the work group with respect to the treatment of choice. Workplace, that is workload, staffing, and infrastructures, that is also important. So these are all factors in information, the scientific information and influence of drug industry. Okay, both are it's the very, very important factor which are influence the use of medicine for the treatment of choice. So these are the factor which are influence the use of medicines. Next thing is the see essential or rational medicines are those that satisfy the priority healthcare needs of the population. Okay, so ultimately our aim or objective is this: it satisfy the priority healthcare needs of the populations. Okay, and whatever they have a purpose, whatever for the sake of the disease, the person or patients are or subject are going to take a medicines. Okay, that should be fulfilled. Okay, it should not be like that the you are taking the XYZ drug for so and so purpose and you are not going to cure. Okay, so they are selected with the due regards to the public health relevance and evidence on the efficacy and safety, and comparative cost effectiveness is very, very important. So ultimately it's matter at all the comparative cost effectiveness with respect to the efficacy and safety and the quality of the drugs. These are the three main pillars of for any medicines or any drugs. So the government of India published the national list of the essential medicine in the year of 2015. Okay, till date, there is no any further revision or revised national list of the essential medicine is not published at all. So there are 376 drugs have been included in that. Devices as well as apart from the drugs, the disposables, consumables, and devices have been included in the essential medicine list. Now, the WHO criteria to guide the selection of the essential rational drug. So here from the clinic, see the how we are going to select the drugs and how you justify with respect to the safety and efficacy of the drugs. That is totally based on the clinical trials, phase 2 and phase 3 clinical trials on animal as well as on the human volunteers. So that safety profile is and efficacy of the drug should be evaluated, taking into the consideration the factors as the treatment facilities available for incidence and pattern of the disease, even the genetic demographic factors, genetic, even though the demographic factors and financial resources of the patients are also important. When more options of the drugs are available under a specific category, the choice should be made on the basis of the relative efficacy, safety, quality, price, and availability. See, there are so many antibiotics are available in the market. Okay, but normally, whatever the doctors what they are doing, okay, the doctors are prescribing very new molecules. Okay, even though the patients are no need that molecules, if the patients are having a very good, it shows a very good effect with the erythromycin, there is no need of to give a drug that is azithromycin. Okay, so once if resistance is developed on azithromycin, erythromycin is ineffective in that case. So the more of the options of the drugs available under the specific category choice, and the it should be made on the basis of the relative efficacy, safety, quality, price, and availability. Choices on the essential drug list influenced by the pharmacokinetic properties and manufacturing, shelf life, and storage facilities. So these are the essential drug list which is influenced by the pharmacokinetic properties. What do you mean by pharmacokinetic property? It is nothing but or related with the ADME process: absorption, distribution, the metabolism and excretion. So that is the choice of on the essential drug list influenced by the why they give more stress on the pharmacokinetic properties. The reason is if pharmacokinetic property is not fulfilled, then it leads to the accumulation of drug in the body, okay, which ultimately very toxic to the human volunteer or patient. Most of the cases, essential drug list only contains single compounds and rarely fixed dose combination product. Okay, so we'll move on and discuss in detail fixed dose combination product in this presentation later on. So most of the cases, essential drug list only contains single compounds, and not the next point is not only should the unit cost of the drug, but also the total cost of the treatment to be considered. Okay, we are not only focused on the single pill, what would be the cost. We are concerned with the all the uh, total cost of the treatments, okay, including the doctor charges, then the nursing charges and everything. Okay, with the advancement of the reliable and accurate drug information, healthcare professional and effective safely utilize the essential drugs. So, with the advent of the reliable and accurate drug information, healthcare professional can effectively, safely utilizes the essential drugs. See, now what do you mean by, see, we are discussing about the uh, કહેવાય કે ભાઈ દવાઓનો વ્યાજબી ઉપયોગ. Okay, now we are moving little bit discussion on the irrational use of drugs. Okay, you all are aware, you are all are registered pharmacists, and you well know regarding the practice, what are the way the combinations are available in the market. Okay, the majority of the combinations, more than 50% of the combinations are irrational. Okay, that we will discuss with a few examples later on. So see, the irrational use of the drugs, the use of the drugs when no drug therapy is indicated. So here the one example is mentioned, antibiotic for upper respiratory infection. Okay, antibiotic, see, it's a viral flu is a viral. Okay, it is not bacterial infections. And like suppose I'm suffering from throat infections, and when I refer any doctors or any general practitioner, what they are doing, they recommend immediately or write immediately azithromycin. Okay, so the use of drugs when no drug therapy is indicated. Okay, so here this picture represents a very good thing towards the quality care together, use of antibiotic wisely. See, if we are not going to care this use of antibiotics, then it leads to the time will come, or it is very near in future, we don't have any new antibiotic molecules. Because right now, if you see in the research pipeline, none of the latest antibiotic available, none of the antibiotic in the research pipeline. So see, after if more and more resistance is developed, okay, in this scenario, then the time will come we don't have any antibiotic for the any cause. Okay, so we must have to use the antibiotics in a very uh, good way. Okay, so like the example mentioned over here, I have a cold, should I use antibiotics? So the doctor always or have to say that the antibiotics are not the right treatment for the viruses such as a cold and flu. Okay, because if you are using excessive or incorrectly, it can stop them stop them from working when you really need. So ultimately the meaning is the resistance is developed over a period of time. So it is very important point. And see, even though the azithromycin, whenever doctor prescribed, they are not telling that you have to take empty stomach. Because see, if you take full stomach azithromycin, 60% of the drugs, 60 to 70% of the drugs are ineffective. Okay, so you are not getting the effect of the exact drug. Okay, so such things are not going to tell by the person or by the doctors. Okay, so the use of unnecessary expensive drugs, the use of the third generation broad spectrum antimicrobial when first line narrow spectrum agent is indicated. So see, if broad spectrum is not required in the treatment, then see, whenever the see the example like meropenem, and these are the categories of the vancomycin, it's too costly. Okay, carbapenem, meropenem, then the fourth generation cephalosporin. See, these are the broad spectrum antibiotics are too costly. Okay, and if the drug is effective by using narrow spectrum agents, then why should we move towards broad spectrum? Okay, because we have to beware with respect to the resistance. Okay, so the use of the unnecessary expensive drugs have to avoid if the treatment is available with the narrow spectrum agent. So we must have to use narrow spectrum coverage, that is penicillin V and G, then lincosamide, that is clindamycin, and the streptomycin, rifampicin, daptomycin. These are all drugs are narrow spectrum. Now the another irrational use of the drugs, that is use of the wrong drug for a specific condition. Okay, which is requiring in the drug therapy. So tetracycline in the child diarrhea required ORS. Okay, so ultimately the meaning of this statement, you cannot recommend, or doctor cannot be recommend the tetracycline in the child diarrhea if the the treatment is available with the ORS. Okay, so use of the wrong drug for this specific condition. The use of the drug with the doubtful and unproven efficacy. If the doubtful and unproven efficacy is there, then the use it is considered as a irrational use of the drug. Use of the drug for the uncertain safety status, like nimesulide. Okay, and failure in the diagnosis. Okay, if you are failed to diagnose the treatment or the any disease conditions, then it is also you are moving towards the uh, irrational use of the drugs. Now the multi drugs use and polypharmacy. Okay, what it means, polypharmacy is nothing but if you need only one tablet, then why doctor prescribe the 3-4 drugs? Okay, there is no need of multivitamins, no need of vitamin C tablet, still they are giving. Okay, then and then we can say it's a prescription of so and so reputed doctors. They are doing a wrong practice. Okay, so multi drugs use and polypharmacy. Failure to provide available safe effective drugs. So failure to vaccinate against the measles and tetanus, or failure to prescribe ORS for acute diarrhea. Okay, so if you have available safe and effective drugs, then why and if the measles or the tetanus is occur in any patient, and if you are not going to vaccinate, then it is considered as a failure to provide available safe and effective drug. The use of the correct drugs with the incorrect administration, doses and duration. The use of IV metronidazole when suppository is an oral formulation would be appropriate and the patient is stable. Okay, there is no any kind of emergency to give IV metronidazole. Okay, so if suppository and oral formulations are available, metronidazole tablet, so why we are using, or why we should give IV metronidazole? Okay, so use of the correct drug, with incorrect administration, doses and duration, that is also important. So use of the fixed dose combination, which are ideal required. C, which is ideally not required, fixed dose combination drugs. See, majority of drugs which is available in our country, uh, uh, see, almost Union Health Ministry has banned 344 fixed dose combination, and a decision would be held by the Delhi High Court. Okay, and see, banned uh, affect the manufacturer and sales of the product worth rupees 5,000 crore. The annual revenue losses at around 4,000 crores. Okay, and FDC, fixed dose combination, is a combination a drug that contain two or more drugs. So what do you mean by fixed dose combination? The drug which contain two or more drugs combined in a fixed ratio in the dose. Okay, and it is available in a single drug. It is available in single tablet. Okay, so if you refer the tablet of diaminic DCA, okay, more than two drugs are there, diaminic DCA. Okay, about 2,500 drugs set to go off from the cells. Around 2,500 drugs set to go off from the cells, and all FDA drugs are not unsafe. Sorry, all FDC drugs are not unsafe when combined with the scientific precision. Okay, they are effective for the patient suffering from acute and multiple conditions such as tuberculosis, malaria, cancer. Okay, so in the malaria and cancer, in even tuberculosis, we need the multiple drug treatment, and it should be prepared with the scientific precision. What do you mean by scientific precision? Scientific precision is nothing but the one drug is compatible with the another drug. Okay, what do you mean by compatibility? The compatibility is nothing but there is no any kind of chemical interaction between two drugs, because if any kind of the chemical interaction is there, then it changes the pharmacological action as well as the drug profile, ADME profile or pharmacokinetic profile of the drug are going to change. Okay, and sometimes the molecule, apart from the safety and efficacy, they become more toxic. Okay, so Indian pharma sector, they are worth rupees 2 lakh crore may suffer in immediate loss of the 1,000 crore, and still another list more than 500 FDC has been sent for the assessment. Okay, till list is not released, and if they are banned, the annual loss occur by our country or by our pharma sectors, that is 10,000 crore. Now, CDSCO, uh, uh, an irrational FDC. So, Ministry of the Health and Family Welfare constituted committee under the chairmanship of the Professor C.K. Kukate, Vice Chancellor of the KLE University, Belgaum, Karnataka, for examining the safety and efficacy of the unapproved FDC, which were licensed by the state drug licensing authority without due approval of DCGI. Okay, so see, total more than 2,500 fixed dose combination were discussed with subject to expert and categorized into the four different category. One is irrational FDC. They are totally banned and immediately they release the guideline to withdraw from the market. FDC requires further deliberation. Okay, there's some drugs out from the more than 2,500 FDC. They put in the category, okay, which required further deliberation. Okay, so they prove the rationality of the fixed dose combination. Then rational FDC, they also categorize rational FDC, excuse me, and they also some drugs are lies under the category of the FDC required further generation of the data and prove the efficacy and safety of that mixture. So the CDSCO is the Central Drug Standard Control Organization. Okay, CDSCO is our Indian regulatory body who approve each and every drug. Okay, so even though one CDSCO approval, then that drug is incorporated in the Indian pharmacopoeia once the profile is fully developed. Most of the FDC have the following demerit. So one is doses alteration of one drug is not possible without alteration of the other drug. Okay, how you can fix in the fixed dose combination, how you can fix the dose of the one drug with respect to the another drug. So that is the main issue or demerit we can say for the fixed dose combination. Differing pharmacokinetic of the constituent drug pose the problem of frequency of administration of the formulation. By simple logic, there are increases the chances of the adverse drug effect and drug-drug interaction, which leads to uh, the toxic effect or increases the adverse drug reactions. Okay, so both the drugs better to take individually. Unfortunately, many FDC being introduced in India are usually irrational. Okay, and example is that the pediatric formulation of the nimesulide and paracetamol. So here the nimesulide alone is more antipyretic than paracetamol. See, it is irrational formulation, irrational combination. Okay, Nimeson P, the very old brand, and it is available in the market, Nimeson P, which is having nimesulide and paracetamol. So here the alone nimesulide more antipyretic than paracetamol, and it is more anti-inflammatory than aspirin. Okay, and equivalent analgesic to any other NSAIDs. Okay, so efficacy gain are unlikely with the added paracetamol. However, the patient may be subjected to increase the hepatotoxic effect from the combination. So that's why it is avoid to use if single nimesulide, if person is going to take, see, nimesulide is it is available in Indian market, but it is banned in US, Canada, Australia, and many more countries. Even the whole European Union, this drug is banned because of the hepatotoxicity. But in India, it is available, and see, it is having a analgesic activity. In India, the variety of NSAIDs combinations are available, often an over-the-counter product, OTC product. Okay, these combinations are an easy way to sell two drugs where one may be needed for the patient. Okay, the combination pills are marketed with this slogan like ibuprofen for pain and paracetamol for the fever. Okay, like Combiflame. Okay, which containing ibuprofen and paracetamol, and ibuprofen for peripheral actions and paracetamol for central action. They are, see, marketing in that way. Okay, thus combining of two NSAIDs does not and cannot improve the efficacy of the treatment. If you combine two NSAIDs, there is no any meaning with respect to the efficacy of the treatment. It is only add to the cost of the therapy. Okay, manufacturer એમના ખિસ્સા ભરે છે આ બધું કરવાથી. Okay, they are increases the cost. Okay, so the only add to the cost of the therapy, and more importantly uh, uh, to increases the adverse effect and toxic effect of the drugs. Combination of NSAIDs and analgesic with the antispasmodic agents are also available in the India. So they are not only irrational but also could be dangerous. The antipyretic drug promote the sweating and thereby help inhibit dissipation means a liberation of the heat or the so once a liberation of heat and sweating occur, what happened? Temperature goes down. Okay, so on the other hand, anticholinergic drug or antispasmodic drugs inhibit sweating. See this combination. Okay, so if you are adding antispasmodic drug with the any NSAIDs, what happened? Okay, one is going to relieve, it promote the sweating as well as it help in heat dissipation, okay, or heat liberation from the body. Then other hand, the anticholinergic, antispasmodic drugs are there which prevent the spasm or inhibit the spasm, okay, that inhibit the sweating. Okay, so combining these two can result in dangerous elevation of the body temperature. Okay, and see, these are the few examples. The enalapril and losartan, both are belongs to the antihypertensive drugs, and both act by the same pathway. So they do not add to the efficacy. Then phenylpropanolamine, which is widely used in the and see this it is even added in the some cup syrup and tablet is also available. So even cold remedy is having a very good application. So part of the many cold remedies, though it is a banned drug and it causes hemorrhage stroke in many patients and it may cause the enlargement of the glaucoma as well as the prostate. So still it is used in India. Domperidone and rabeprazole, it is also irrational combination. And see, there are 344 combinations are there, which are irrational, and it is published by the National Health Portal. You can, it's a government portal, you can refer it, and I will show you at the end of this presentation. Okay, the National Health Portal, all 344 combinations are given which are irrational. Okay, so ultimately it increases the risk of rhabdomyolysis. Rhabdomyolysis, that means the breakage of the skeletal muscles. Okay, which ultimately it leads to even one of the cause of the kidney failure. And see, I had taken a screenshot from the CDSCO site, that is Central Drug Standard Control Organization. And see, the list of banned drug is also there. And release the date is 1st January 2018. This you can also download from that, the banned drug in India. Now impact of the irrational drug use. So see, whatever the irrational use of the drug, what is the impact? See, it impact on it every where with respect to our life cycle. See, it reduces the quality of the therapy. Okay, irrational drug or inappropriate use of the drug, it reduces the quality of the therapy, which leads to morbidity and mortality. Sometimes it is life threatening. Waste of the resources which required availability and increase the cost. Okay, so there is the waste of resources, risk of unwanted effect, so adverse drug reaction and bacterial resistance, that is risk of the unwanted effect. Okay, and psychosocial impact, that is patient rely on unnecessary drugs. Okay, so these are the inappropriate use impact on the irrational. So main it is categorized, or mainly it is there are four factors: it reduce the quality of the therapy, waste of the resources, risk of the unwanted effect, and psychosocial impact. So WHO says that the antimicrobial resistance is one of the world most serious public health problem being faced. As I said, right now in the research pipeline, none of the antibiotics we have for the next 20 years. Okay, because once, see, discovery of the molecule from the very first step to the last step, it requires 15 to 20 years. And for the next 20 years, we don't have in the research pipeline any new antimicrobial agents. So that is the, see, WHO give very high emphasis on that. It gives very stress on the antimicrobial agents, that the to prevent or to reduces the unwanted use of the antibiotics. A major reason of the irrational use of the medicine, according to WHO, worldwide, more than 50% of the all medicines are prescribed, dispensed, and sold inappropriately, and 50% of the patients fail to take them correctly. So 14th WHO model list of the essential medicines contain only 18 approved drug combination. See, only 18 approved drug combination. See, if you see in US or any developed country, they are not allowed fixed dose combination. Whatever tablets are available, a single tablets are available. Okay, they are not allowed only if the US FDA accept that thing, Okay, the scientifically approved and valid combination, they are approved, otherwise they are not entertained at all fixed dose combination. Okay, whereas in India there are innumerable, okay, we cannot calculate even that the how many number of the uh, irrational drug combinations are available, we can say, which are available and can bought without necessary giving, and you can easily purchase from anywhere, from any medical store. So see, even though the skin, if I'm talking about the skin treatment, see, so many antifungal drug combination with antifungal drug with these different steroids are available, which are all are the irrational combinations. Okay, the factor underlying irrational use of the drugs. So the major forces can be categorized as those deriving for the patient prescribers and the workplace and the supply system, including industry influence, regulations, and drug information and misinformation and combination of these factors. So these are all factors, or these are the major forces which are responsible for underlying irrational use of the drug. ઓકે સપોઝ મારા કોઈ રિલેટિવે કોઈ મેડિસિન્સ કોઈ ડૉક્ટરને બતાવ્યું હોય તો એ એની ફ્રેન્ડને બી બતાવશે કે ભાઈ તમે આ લઈ લો ચાલી જશે, તમને આ જ પ્રોબ્લેમ છે, મને બી આવું જ હતું. So this is the irrational use of the drug. We don't know the the overall body response with respect to drug with the another person. Okay, so we cannot even though suggest the same drug to that your friends or your family members. Okay, sometimes some drugs are allergic to one person but it is not allergic to other person, so which create a life threatening issues. Okay, so major forces can be categorized uh, uh, as those deriving from the patient's prescribers, workplace, supply system, including industry influence, regulation, drug information, and misinformation and combination of these drugs. So prescribing, number one. Now, what are the underlying irrational factors for prescribers? Number one is lack of education and training. Okay, lack of objective drug information, generalization of the limited experience, misleading belief about drug efficacy, and wrong diagnosis, which is very horrible things if wrong, if you the doctors wrong diagnose the disease, then it is uh, the life threatening condition may patient would be there. So the lack of education and training. So that's why, see, for the prescriber, for the doctors, each and every pharma industry, every over a period of time, they are organizing CME. All the MRs, those who are in the refresher course, they are aware about the CME. Over a period of time, they are arranging CME. So the doctors are aware with respect to the new drugs and new therapy, whatever upcoming in the market. So now the patients. So patient, that drug misinformation, misleading belief is there. Patient demands and expectations are there, and there is a see, there is nothing like that. There is a pill for every ill. Okay, some things, many things are psychological. Many things are psychological. Okay, so see, even though the patient suggests, ડોક્ટરને જ્યારે પેશન્ટ મળવા જાય ને ત્યારે ઘણી વખત એવું થતું હોય કે ડોક્ટરને સામુથી પેશન્ટ એવું કહે કે સર મને બોટલ ચડાવી દો. Okay, that is the misleading belief of the patient. બરાબર, એને વીકનેસ લાગે છે, ઇટ્સ ઓકે, દેટ ઇઝ બીકોઝ ઓફ ધી સમ ડિસીઝ ઓર સમ ધી મેડિકલ ઇશ્યુઝ. ઓકે, તો ડૉક્ટરને એ સલાહ આપશે કે સર મને બોટલની જરૂર છે, ગ્લુકોઝની બોટલ ચડાવી દો. ઓકે, એન્ડ ગૂગલ ભગવાન તો છે જ, ઘણા બધા લોકો તો ગૂગલમાં સર્ચ કરે, બટ ઇટ્સ સી the very bad practice, or we can say you cannot rely on that resources. Drug regulations, the unreliable suppliers, then drug shortage, expired drug supplied. So these are the drug regulation, industry promotional activities, and misleading claims. Okay, misleading claims, we are observing this so many drugs, they are mislead. Okay, especially in cosmetic treatment, I say that it's a practical example. You all are aware with the nose car. Nose car is one of the cosmetic fairness cream, and see, it is proved, but still government is not going to take any kind of the action against them. They are adding steroid in that for the subside or for the removal of the pimples from the face. So I had discussed this thing with the one of the skin specialists of the Karamsad Medical College, and she also told me the same things. It is avoid to use by the the personnel, because no scar cream they are adding steroid in that. And see, it is not written in the ingredients list, but it is proved. And they also submitted the data to the Indian Drug Manufacturing Association and even CDCO, but still it is under process. Okay, in upcoming future they are going to take government take some action against them. Drug supply system, the drug shortage, expired drug supplied and unreliable suppliers, workplace heavy patient load, pressure to prescribe, then lack of adequate lab capacity, insufficient staffing. These are the all workplace. Next is the factors which demand the rational drug use. Drugs explosion, antibiotic resistance, increase the cost of the treatment, growing awareness, Consumer Protection Act. So these are the factors which demand the rational drug use. Okay, so factors which are, see, one by one I'm going to take in a brief. So drug explosion means you have more number of the drugs are available, and the incredibly complicated the choice of the appropriate drug for the particular indication. So that is drug explosion. So you have so many drugs for the particular class, like in NSAIDs you see the aceclofenac, the naproxen, there's so many molecules are available, but which molecule is appropriate to the particular patient. Okay, that is very important. Efforts to prevent the development of the resistance, that is for the antibiotics. Okay, so overuse of antibiotics we have to prevent. Growing awareness today, the information about the drug development, it uses adverse effect, travel from the one end of the plane to the, sorry, the adverse effect. See, these are all things, the whatever the information we have with respect to the development and uses and adverse effect, it should be traveled from one end of the planet to the other, and within with an amazing speed through our various media. So see, the scenarios of social media, and even the Google Bhagwan is also there, and everything they are saying, okay, you just have to refer it. The patients are doing this thing, okay, and they take own decision which is very risk with respect to that person. Okay, even though they also interpret the blood report through the Google. So increase the cost of the treatment, increase in the cost of the drug, increase the economic burden on the public as well as on the government, and this can be reduced by the rational drug use. The Consumer Protection Act, see, this is very important. Okay, and Consumer Protection Act, the for the medical profession, they may restrict the irrational use of the drugs. Some example of the commonly encountered inappropriate prescribing practice in many healthcare settings include overuse of antibiotics, indiscriminate use of injection in malaria treatment, multiple and overprescription, okay, excessive use of the antibiotics for treating minor infections, use of irrational drug combination for treating cough, multivitamins, tonic for the malnutrition, okay, and unnecessary uh, use of the expensive antihypertensive drug. Okay, so these are all uh, uh, we can say encountered in points which are encountered with respect to the inappropriate prescribing practice. Okay, where multivitamin patient is very healthy, there is no need of multivitamins, there is no need of the tonic. Okay, and still doctors are prescribing the things which are, we can say, unethical practice or irrational things. Now, the WHO conference of the expert in Nairobi 1985, they are defining the rational use of the drugs, okay, for an adequate period of time. Rational use of the drug for the adequate period of time. The, see, this point is very, very important, especially those who are taking antidepressant drug or those who are taking an antipsychotic drug. See, the patients have, see, normally doctors have an intention, once the treatment is going to start with the any antidepressant and antipsychotic drugs, it's a lifelong. It's a lifelong. Doctor is not in have an interest to counsel them and to reduce the dose, and ultimately he or she is not in interest to withdraw the drug from the prescription. Okay, so these are the things are there, and patient build a tendency that uh, they have to use this drug, okay, for a lifelong. Okay, but it should not be like that, especially in antidepressant and antipsychotics. Patient receive medication appropriate to their clinical needs, doses that meet their own individual requirements and at the lowest cost to them and their community. So these are all important points. The appropriate medication should be prescribed Then doses, whatever you give to the patient, it should be meet their own individual requirements and at the lowest cost. Okay, cost is also matter at all. Okay, next is the benefit of the rational use of the drugs. So we are, see, if we are giving a more stress on the rational use of the drug for the better community healthcare, and ultimately our goal is to improve the quality of the life. Okay, by the or through the attainment of the highest possible level of the health. Okay, so there are five rights: right patient, right drug, right dose, and right route. Even though time, so time is also important, route is also important, dose is also important. And why time is mentioned, you have to say to the patient or person, okay, you have to take with empty stomach or full stomach or with meal. Okay, so these are the very important thing. Even though you have to take at the bedtime or the early in the morning, the things are which is very very important. So critical factors which are there for the choice of the medications, and see, as I said, the three important pillars are there for the choice of the medicines, that is safety, efficacy, quality of the drugs. And fourth one is there, that is the cost treatment, that is cost benefit ratio. Now the requirement for the rational use of the drugs, and in that the very first thing is the adequate diagnosis. See, first two points are concerned with the doctors. For the correct diagnosis is very important, and the prescriber must have adequate knowledge and motivation. The even though they have the examination facilities, sufficient time to take sufficient time to take history, as well as the explain to the patient or a diagnosis of the treatment. Correct prescription is very very important. Okay, because see, if the prescription is not correct with respect to the diagnosis, or if diagnosis is not adequate or improper, then everything is going to fail. Okay, we are the doctors are not common conclusion, and patients are not get relief from the particular problem or particular disease conditions. Appropriate dispensing, for the correct dispensing to occur, a dispenser must train. See, it is concerned with the pharmacist. Must train, have adequate time, and have the necessary materials like container, labels, and everything, and have a dispensary where the possible to communicate with the patients. Okay, see, that is, see, already JK sir covered the patient counseling, which is a very very important part. And the important thing is, if you make a counsel to the patient, you can increase your business. Okay, because see, once patient reach to you, if you say that, okay, don't worry, everything is under control અને બધું સારું થઈ જશે, એટલું તમે કહો ને, ત્યાં પેશન્ટનું અડધું દુઃખ ઓછું થઈ જાય. ઓકે, અને તમે એને કાઉન્સલ કરો, યુ જસ્ટ કાઉન્સલ ધેમ, ભલે બહુ સિરિયસ કન્ડિશન હોય. Okay, so he relieved from the tension or pressure of the disease. And the second time, the prescription should be repeat at your own medical store. So it is a very good thing, patient counseling. Patient adherence to treatment, which is very very important. If you counsel them very well, then and then they adhere to the treatment, is dependent on the understanding and acceptance of the treatment. Okay, if you are not counseled well, patient immediately refuse and stop the medication. Okay, like if see, in our country, majority of the peoples are illiterate. Even literate people are also do the things. Okay, if they are taking antibiotics for this so and so reason. Okay, and before completion of the full profile of the drug treatment, okay, based on dose leader, if they feel very good, okay, they immediately stop the medication. So what happened after the 1 week, relapses occur, reoccurrence of the same infection. Okay, so these are the things which is very very important, especially these things are happening with the in the urinary tract infection. Okay, so patient adherence to treatment is dependent on understanding and acceptance of the treatment, and this results for effective communication between, see, over here the doctors, pharmacists, and patient all are have give more stress with respect to the patient compliance. See, these are the few examples. The drugs which they see the labels, almost they are having the same brand name, but the content are different. Okay, and it occurs the medication error. And that's why in the year of 2016, Supreme Court says that every doctors have to write the only the name of API, only the name of active pharmaceutical ingredients in a capital letter. Okay, but still doctors are not following. They're writing a brand name. Pharmacists decide which brand recommend to the patient. Okay, so see the medazole, itraconazole, antifungal drug, esomeprazole is there, same brand name. Okay, and it's antacid. Okay, and another is albendazole and antihelminth drug is there. Then pantoprazole, antacid is there. Okay, then metronidazole. See in the below, the spot the difference. Even though labels are also same. Okay, metoprolol tartrate, the same packaging material, secondary packaging material is same. Okay, and azathioprine, metoprolol is used in the treatment of the hypertension, while azathioprine it is a immunosuppression drug. It is widely used to prevent the rejection of the organ transplantation. See the medication error with respect to the labeling of heparin, sodium, and midazolam. See gabapentin and gamfibrozole. Okay, so which create the labeling and colors and everything would be same in the on the labels, which mislead and it leads to the medication error. Now, what is the role of pharmacist? You all are aware, you are all are involved in the day-to-day work, the role of pharmacist, but still it required the things where the some areas where you can improve your business, even though you can improve the your way of communication towards the patient. So patient counseling, inventory control, then drug procurement, refiling of the prescription, discourage the self medication. So self medication is another one big issue. As I said, I have for so and so course, if I refer a doctors and the another doctors are going to means I suggest the same drugs for the my friend or somewhere else my family members, it is not a good practice. Excuse me. Do not sell medicines without prescription. Okay, so this is all about the role of, you all are aware and you are all are working in the different areas. See what to do or what not to do. So see the no self medication, no own antibiotic kit. Okay, emphasize on dose and duration. Educate people around you. You have to educate and stop them. See, right now in the COVID pandemic, okay, the pharmacists play a very good role. Some of the friends who are retail pharmacists, they are doing a very good thing to counsel the COVID-19, or those who have some symptoms with respect to the uh, like the breathing issues and some throat infections. They're counseling well. Okay, don't worry. You just counsel them and boost confidence. Okay, that is very very important. So we have to avoid the self medication and not lies without showing without consulting a doctors, and you are taking your own way antibiotics. That is very bad practice, and you have to do just educate the people around you. Okay, so see this is the uh, what happened exactly. This the retail pharmacist story is there, and first of all you have to see the validity fulfillment of the legal requirement for the medicine to be dispensed. Okay, then we have to move on the next thing, that is the patient details, age, indication of the medication, current medical conditions, etc. Then you have to move towards the appropriateness of the prescription medication, that is type, dose, strength, frequency, and duration. Then potential drug interaction, does any kind of drug interaction is there, so that is very very important. Okay, does it interact with the other concurrent medications? Okay, so many hematinic drugs are there which is not recommended at all with the some of the anti-malarial drugs, because they're having make a chelation or complex formation occurs, which ultimately leads to accumulation of drug inside the body. So that we have to see, or so potential drug interactions, uncertainty or error detected. If yes, if anything is found uncertainty or error, immediately you have to consult with the doctors, and new prescription is required. If there is no any kind of uncertainty in error in above 4 points, then you keep record keeping, label printing, and filing the prescriptions, and you can dispense and counsel to the patient. So better pharmacist means a better healthcare system. Okay, thank you very much, everyone. Okay, if you have any question, you can ask me. So some patients are going to take.
Questions? Yes, sir. Some questions are there
Yes.
with us from the participants. Okay. First question is, does the antibiotics or antivirals have the side effect of coagulation?
See, normally the antibiotics and antiviral are not having any side effect with respect to the coagulation. Okay, it depends. See, coagulation is totally depend on the level of calcium in the blood. Even though the level of vitamin K, it is a act as a coagulation factor. Vitamin K is very important for the coagulation of blood. But antibiotic and antiviral drugs are not having such kind of effect.
Okay. One more is there. How does the efficacy affects the rationality of drug?
Yes, see, if I compare I would like to give an example. The drug like if you see nimesulide, and or I can say that the uh, diclofenac sodium, and if you see the drug of aceclofenac. See, aceclofenac is one of the recent molecule which is very good efficacy. See, efficacy is direct concern with the potency of the drugs. So how we can reduce the dose of the drug, and by using the or by exposing your body with a minimum dose of the chemical moiety with a more safety profile, then it is very good way to select the drug. Okay, so over here, as compared to diclofenac, I can say that the aceclofenac is more safe, because if you see the dose profile, it's having a wider difference. Okay, so we can get the better effect with the minimum chemical exposure of uh, particular API.
Okay. Sir, one more is there. What is the way forward or action GSPCs should take to transfer the information to the pharmacist when there is a recently found drug incompatibility or generation of unknown impurities which are very toxic to the patient?
See, GSPC have the DIC center, okay, Drug Information Center, and see, if you have any kind of excuse me, if you have any kind of doubt, even though if you find something on the internet, you can also communicate, because their experts are there, and they are also concerned with the different government hospitals, and they are also collecting some important data with respect to pharmacovigilance, where they are reporting the adverse drug reaction of already existing molecules, whatever the older and new molecules are there, they are using, or they are sharing the information with you, all of you. So the number is available on the State Pharmacy Council website. Anytime you can contact to the DIC, even though you can drop the email, within 24 hours you can receive a particular information whatever you require. And see, I would like to show us a slide, sorry, internet screen that which is having a list of drug which is banned, or irrational combination of drug, we can say.
Sir, one last question is there.
See, hope it is visible to everyone.
1 second. Yes, it is visible, sir.
So here, see, there are 344 drugs are available. Okay, it is a complete list of the drug combination which is banned by the ministry. See, these are all combination. So many examples are that: aceclofenac, paracetamol, and rabeprazole combination, nimesulide, diclofenac, then nimesulide, cetirizine. There are so many drugs are available, and you can refer just National Health Portal. National Health Portal. You can just write in Google and you can find this. It's a published thing. Yes. Yes,
Uh,
ma'am.
so last question.
Yeah.
In today's pandemic condition, use of multivitamin has been increased a lot. So is it safe to take it?
See, if patient is healthy, I said better to, instead of multivitamins, better to lie on the fruits, especially citrus fruits, because vitamin C, which is very helpful to boost the immunity, even though the immune boost, whatever fruits are available in the seasonal fruits, you can consume. It is better to take rather multivitamins.
Okay. Thank you so much, sir, for accepting our invitation, first of all, and for sharing your reflective knowledge with us, sir, today.
Yeah,
Thank
thank you
you.
very much.
Thank you. Moving towards the last session, we are having with us Dr. Sandeep Patel, Assistant Professor, LM College of Pharmacy, and he will be delivering a talk on interpretation of lab data. I would request Mrs. Rishita Patel, Assistant Professor, ISCP, to introduce Dr. Sandeep Patel, sir. Over to you, ma'am.
Thank you, ma'am. Thank you, Heta, ma'am. Good afternoon to each and everyone. I would like to introduce Dr. Sandeep Patel, sir. Sir is currently working as an assistant professor in a pharmacology department at LM College of Pharmacy. Sir has done B.Pharm and M.Pharm in pharmacology in 2004 from LM College of Pharmacy, Ahmedabad. Sir has done his PhD in 2011 from LM College of Pharmacy, Ahmedabad. So has been awarded certificate by WIPO, International Academy for successfully completing course in intellectual property right in May 2006. Sir has received AICT fellowship of rupees 90,000 for his M.Pharm. Sir has successfully completed Diploma in Pharmaceutical Production Management from IPER, Pune. Sir has more than 9 years of experience in teaching in the areas of pharmacology, toxicology, reverse pharmacology, development and validation of experimental animal models. Sir has published more than 21 research papers in various national and international journals. Sir has presented more than 9 posters of his research work in various national and international seminars. And sir is going to deliver a lecture on interpretation of a lab data. I am heartily welcoming you, sir. I am handing over the session to you, sir. Over to you, sir. Thank you, sir.
Am I audible?
Yes, sir, you are audible. We can start with the session now.
Okay. So I'm sharing my screen. Uh, okay. Is the slide is visible to all of you?
Uh, yes, sir, it is visible.
Okay. Fine. Uh, so first of all, good afternoon to all the participants, management, respected Harsha ma'am, and faculty members. I would like to thank you, the management of IICP Charutra Vidya Mandir, for providing me the opportunity to talk on this 2 days refresher course for the registered pharmacist, GSPC sponsored course for registered pharmacist. So today I'm going to talk on interpretation of laboratory data. Okay, so whenever the laboratory data comes into the picture, we are always looking at the value which are deviating from the normal. Whenever we come to the particular thing, as I say, if I'm handling any laboratory data, I'm always looking at the reference level and how the my value or the patient value is deviating from the reference range. Okay, and based on this, we are assuming certain disease conditions. We are always looking the signs and symptoms of the patient, and we try to correlate those signs and symptoms with the patient's patient's disease conditions. So now certain important aspects I would like to highlight before we go to the individual laboratory data. So most frequently used test for the laboratory data, they are complete blood counts, electrolytes, blood chemistries, and urine analysis. They are broadly classified. These laboratory data are broadly classified into these 4 categories. Now, when we are interpreting laboratory data, certain things that we should know, we must know before we come to any conclusion. Whenever we are interpreting lab data, we are always looking at the normal values, but these normal values may vary from laboratory to laboratory and depending on the techniques and reagents which are being used for the estimation of that parameter. There are several examples which are related with these values. Okay, so, one of the example is glycated hemoglobin. Previously, I observed many deviation in the reference range from lab to lab also. So always each and every laboratory has to decide their own normal value. Otherwise, in most of the routine biochemical estimations, we are having reference range and that they do not vary from lab to lab. But in some of the specialized tests, we may see the deviation in the reference range. We may see the deviation in the reference range. Now, the normal values, they also may vary depending on the patient's age, gender, weight, height, and other factors. There are several examples. I would say, if I say the neonates, take an example of bilirubin. The bilirubin level, if we talk about the bilirubin, the normally the bilirubin level is elevated in neonates. Okay, because when they come into the external environment, there is a massive rupture of the RBC, and bilirubin level is greatly increased in those in neonates. So, but as the age progresses, the bilirubin comes to the normal. Okay, so the age is the factor. Gender, we always see the difference in the hemoglobin level between male and female. Weight, height. There are several factors which may affect the patient's laboratory data or particular laboratory test. Now the laboratory error is, hello, moderator, please. Someone is raising the hand. Is there any question if I need to highlight? Hello.
Sir, we have opened the question and answer box for any questions if the participant have. I request the
Okay.
participant to drop the questions over there. Okay. Sir,
Fine.
you may continue.
Okay. So there are all chances of the error in the laboratory investigations, and this error need to identify. Okay, there are several types of the error which may happen, like technical error, improper calculation, inadequate specimen, incorrect sample timing, improper sample. I'm just giving one example for each instance. Technical error, say for example, if I'm collecting the blood sample for the estimation of creatinine, and instead of that, i'm I'm collecting the blood, or rather the phlebotomist which collect the blood very wrong way, and if there is a problem in the separation of the serum or plasma from the blood, there may be a possibility to change in the creatinine value. Improper calculation, always dilution makes the difference a lot. Many times, if we are identifying the urinary sodium level, and if dilution is not properly done, we may end up with the elevated sodium level, and many reports are suggested such kind of incidences. Inadequate specimen, again the blood sample or the volume of blood or volume of this particular specimen is not corrected in appropriate amount. Incorrect sample timing, this is something important when we are estimating the enzymes, because the enzymes are released from the sample after some time, and if we are immediately withdraw the blood after having any incidents, the enzyme doesn't get released from the damaged tissue. So we may end up with the false negative result. And sometimes and these enzymes, they are again wiped out from the body within 24 to 36 hours. So even if we measure the enzyme concentration after a certain amount of time, we couldn't get any enzyme in the blood sample, so that end up with the false positive. Okay, so these are the two different extreme in which the laboratory errors are likely to happen. Improper sample. Again, as far as several incidences, just look into the recent situation, the COVID-19. The scientists tried various samples for the estimation of the COVID, the rapid identification of the COVID. Okay, and they end up with the conclusion that bronchoalveolar fluid is the most appropriate sample for the estimation of COVID-19 virus, for the detection of COVID-19. Okay, even though there are certain research paper which suggested that the nasal or the nasopharyngeal or oropharyngeal swab may not contain virus. There are 85% accuracy when we collect the swab for the COVID-19. Okay, so bronchoalveolar fluid is having 100% sensitivity. In contrast to that, even though the blood is collected for the antibody detection, that may only shows the 50% sensitivity. On the other hand, there is an example of urine collection. Urine won't display any COVID-19 traces. So if we collect the urine, then we couldn't find anything about the COVID-19. So the collection of the appropriate sample is very much required. Okay, another example I am giving you, the leukemia. If we want to identify the leukocyte infiltration in the brain, we must collect the cerebrospinal fluid. Okay, we couldn't get anything if we measure the leukocytes in the blood. It shows the normal count or elevated count, what is usually happen in leukemia. But if we want to see the infiltration of the leukocytes in the CSF, we need to collect the CSF only. Then the preservation is also a problem. Sometimes if we are measure the hemolyzed blood, the potassium is released in extreme amounts, so we may end up with the false positive result. Okay, because most of the potassium is residing in the RBC. So if we measure the potassium level in hemolyzed RBC, then it won't have the correct results. Food substance is also affecting the specimen. Many a times the lipid level is always measured in the fasting conditions. So because the food may interfere with the correct diagnosis of the lipid related parameters, and medication interference with the laboratory test. This is something very interesting to know about the pharmacist, and where we pharmacists play a major role. I'm giving you an example of rifampicin. The rifampicin shows the orange red coloration of the urine. Now, whenever it appears to the orange red color to the urine, it shows the hematuria. Many patients may have fear that I may have hematuria. That means the presence of blood in the urine, but it is not true. It is drug induced change in the color. Okay, so as a pharmacist, we need to guide the patient. This is a purely a drug related phenomena, and many a times it happens that the patients stop taking drugs for such incidences. So as far as laboratory data they are concerned, our major role is to interpret the laboratory data in a much sensitized way. Okay, we are many a times it observed that we may not correlate the laboratory data with the patient signs and symptoms. Okay, or sometimes we also observed that the patient's laboratory data is normal, but patient shows the signs and symptoms. So at that time, the diagnosis is very much difficult. We also see the situation where patient won't show any signs and symptoms, but patient is abnormal if we look at the laboratory data. So before we go to the any particular step, what we are looking for, we must correlate patient signs and symptoms with the laboratory data, and we do not always rely on the single parameter. We need to take a series of tests in order to identify the particular disease condition. If laboratory error is suspected, the test should be repeated. Okay, one should not, and many times we observed it, that if there is any error in the estimation of the parameter, the laboratory may try to hide such kind of cases. But this is not a ethical approach. So remember, always treat the patient, not the laboratory value. Many a times trying to we are trying uh, to to reduce the laboratory data, but that is not true. We always need to treat the patients. Always need to treat the patient's signs and symptoms. We are not always inclined to treat the laboratory value. Okay, so furthermore, consideration for the laboratory data, the reference range to be quoted for individual test, and it is always there. I rarely find any laboratory who doesn't mention this reference range into the in their report. Based on the data obtained from a sample of the general population served by the testing lab, which is assumed to be from healthy and disease-free individuals. So whatever reference range it is derived, it is from healthy and disease-free individuals. Okay, so if I considering the statistical values, the many test value have normal distribution, and the reference values are taken as mean plus or minus 2 sg. Okay, and that include 95 percentage of population. Many of you may know the statistics. Always there is a bell-shaped curve when we are plotting the graph of laboratory value. That means there must be a symmetrical distribution, and the specific range we can come to know. Say for example, the blood glucose level, it must in between 70 to 110 milligram per deciliter. Now how this value that come into the existence? That means the patient remains normal during this reference range. If anything that deviate, that may suggest any kind of the disease condition or onset of the disease. If I say the blood glucose level is 115 milligram per deciliter, and I wouldn't be able to say that patient is having diabetes mellitus and we need to start the oral hypoglycemic agent. This is not true. We always need to correlate the patient signs and symptoms. Okay, and the patient termed to be pre-diabetic, so we can have certain advice to the patients to have the lifestyle modification so that blood glucose level that goes to the normal. uh, a series of values, rather a single or simple test value may be required in order to ensure clinical relevance. Okay, and to determine or to eliminate the erroneous values caused for example, the spoiled specimen or interference from the diagnostic or therapeutic procedure. Many times we see this particular uh, 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 situation, and many a times I'm just taking an example of uh, of the patients with the gastroesophageal reflux disease conditions. The patient is taking pantoprazole for almost 6 months, and the doctors are still, the patient is having the problem of hyperacidity, and the doctor advised to have the gastrin level, and the laboratory failed to recognize the interference of proton pump inhibitor. The gastrin level is severely elevated. and that may suspect the different disease condition, and that may require the series of tests to diagnose the particular disease conditions. But if on a very first instance, if we identify the drug interference in the laboratory investigation, we should eliminate it. Say again, take an example of aspirin. The aspirin would interfere with the prothrombin time, the bleeding time. Okay, so if we want to measure the bleeding time, at least we should discontinue aspirin therapy for 7 days. Then and then your prothrombin time would be better. That comes true. Otherwise, there may be a problem in the prothrombin time. Okay, or bleeding time, I would say. Okay, so many a times the spoiled specimens, as I mentioned earlier, the potassium level, which is restricted to the RBC, and the hemolyzed specimen of hemolyzed specimen, if we investigate it for the potassium, it would turn into the wrong result. Okay, so furthermore, a disturbance of one parameter often cannot be considered in isolation without looking at the pattern of other tests within the group. Just take an example of creatinine kinase. Okay, creatinine kinase, CK, that is usually elevated in the event of acute myocardial infarction. It is also elevated in case of the muscle rupture or muscle wasting procedure, muscle wasting conditions. Now, this creatinine kinase, if you measure in isolation, and if the level is elevated, you may have the dilemma whether the patient is having some acute myocardial infarction or muscular dystrophy. Now, for if we want to identify the acute myocardial infarction, the patient need to have the ECG, and if after that we can identify the ST segment elevation. So whether it is STEMI, the ST segment elevated myocardial infarction or not, that we come to know. But the alone creatinine kinase won't give you any result. They say, for example, another parameter, that is gamma glutaryl transaminase enzyme, that is known as GGT, which is getting elevated in if the patient is taking alcohol. Now, even though the single parameter which is elevated, we may not say that the patient is having severe acute alcoholic fatty liver disease. It is not true. We need to measure other parameters before we come to the any conclusion. So what I mean to say that the laboratory parameters are always considered as a as a as one of the parameter which used to diagnose the particular condition, but it is not a pure indicator for the particular disease condition. Just it helps to identify the conditions. Okay, in upcoming slides now we are going to discuss various laboratory parameters, namely they are hematological analysis, electrolyte estimations, biochemistry, and some of the sero related estimations, antibodies related estimations. As this is a very long topic, I may not be able to finish it in a very specific time limit. But I will try my best to finish the most of the parameters, as this is not a 1 hour discussion for the laboratory parameter, because numerous laboratory parameters. I'm going to just discuss few of the routinely analyzed laboratory investigations. I'm not in a position to cover all the laboratory investigation in 1 hour, so pardon me for the same. And if you have any question irrespective of the parameters which we discussed in this 1 hour, you may ask in the question answer session also. I will try my best to answer all of them. So let us start with the first, that is complete blood count or the hematological parameters. Most of you are knowing it, and it's a very first step to identify any particular disease conditions. And I hope most of you, I believe that most of you undergo this particular investigation once in a lifetime. So hemoglobin, hematocrit, white blood cells, red blood cells, red cell indices like MCV, MCH, MCHC, platelet count, WBC, differential WBC. So let us start with the first part, the red blood cells count, the RBC count. The normal value is 4.2 million to 5.9 million cells per cubic millimeter of blood, or female it is 3.5 to 5.5 million cells per cubic millimeter of blood. So usually this RBC count is elevated, and that condition is known as the erythrocytosis, which is associated with the polycythemia vera. It is also increased in the individuals who are residing at the higher altitude, and it gets elevated during this strenuous exercise also. Now, even though this is strenuous exercise, if you see the elevated RBC level, we may not come to the any conclusion. Okay, polycythemia vera, that is RBC cancer. The elevated RBC level or as a kind of the RBC cancer, polycythemia vera. In this condition, blood gets viscous and it gets stuck into the minor capillaries, and that may provoke the congestive heart failure or sudden heart failure. Reduced RBC mainly observed with the anemias, lymphoma, and leukemia. The anemia is very much known to us. Lymphoma and leukemia, that is a kind of the liquid cancer uh, where we see the progenitor cells fail to differentiate into their mature cells. Okay, usually the immature cells હોય છે blood cells એ એના routine cells ની અંદર જે mature cells માં convert નથી થતા. That is the major reason why do we observe the reduced RBC. After puberty, the females have lower red blood cells and hemoglobin due to the abnormal or due to the menstrual bleeding, and this is one of the reason. Another reason that would say the hormonal difference, the testosterone, the male hormone, which promote the release of erythropoietin from the kidney, and that is usually responsible for the elevated RBC level. Reticulocytes, they are non immature RBC. Its range is 0.1 to 2.5 percentage of the RBC. It is non-nucleated RBC. So many a times the increased reticulocytes observed with the hemolytic anemia, or a hemorrhage, or sickle cell disease conditions, where there is a abnormality in the RBC shape. It gets a sickle shape. So in that part, the reticulocytes counts are increased. The reticulocytes are also they are indicator of the response of the treatment, which is usually happen to the anemia with respect to the iron, vitamin B12, and folate deficiency. And the reduced reticulocytes, they are always observed with the infectious reasons, alcoholism, renal disease. As I said earlier, the chronic renal failure, where the kidney fails to produce the erythropoietin, and that turn into the reduced red blood cells. Untreated iron deficiency anemia or drug-induced bone marrow suppression. Hemoglobin level, again, 14 to 18, and in female it is 12 to 16. So if we see the structure of hemoglobin, there are 4 polypeptide chains. There are 2 of them are alpha and 2 of them are beta. There is a central it consists of the 4 peptides and having the iron, that is ferrous and protoporphyrin, and this iron carries the oxygen, and that's why the hemoglobin is having the oxygen carrying capacity. So the elevation of hemoglobin is consistent with the RBC, as we discussed earlier. So it is also observed in the chronic obstructive pulmonary disease conditions, and sometimes the chronic smoker may have elevated hemoglobin level, and higher altitude and strenuous exercise are the reason for the elevation of hemoglobin. The reduced hemoglobin that is always observed in the iron deficiency anemia, where the iron won't incorporate into the hemoglobin synthesis, and thus it turns into the hemoglobin deficiency. And most of the Indian populations are having this kind of anemia, that is iron deficiency anemia. The hemoglobin reduction in the hemoglobin is observed in blood loss hemolysis, that is rupture of RBC, pregnancy, fluid replacement, that is as I say, it's called blood dilution. Okay, it is known as the dilutional anemia. Okay, so if we are given higher fluid intake to the patient, this rarely happen, it won't happen. But if there is an elevation of the blood volume, the proportionate RBC level or hemoglobin level is decreased. This is another parameter which is measured is hematocrit. It is known as the packed cell volume, and it is the volume of blood which is occupied by the RBC. Okay, and this hemoglobin, hematocrit, its increased level and decreased level is almost consistent with the hemoglobin and RBC. It is much more important to describe this level, especially in anemias, blood loss, pregnancy, cirrhosis, that is liver related phenomena, liver inflammation, hyperthyroidism, and leukemia. Then MCV, that is mean corpuscular or mean cell volume, that is 76 to 100 micro millicube per cells, so or 70 to 100, 76 to 100. So this MCV, it suggests the size of RBC. If MCV value is increased, that is known as the macrocytic, and if it is decreased, it is known as the microcytic. There are certain normal RBC which is having the MCV, but that comes into the normal range, so it is known as the normocytic RBC. The increase in mean cell volume is usually associated with the folate deficiency and vitamin B12 deficiency. Why do we need to measure this parameter? It is required to identify the megaloblastic anemia or pernicious anemia. Okay, if we have the hemoglobin level, we come to know it is anemia. If it is reduced hemoglobin, we come to know it is anemia. But the identification of this parameter would help us to know the specific type of anemia, whether it is iron deficiency anemia or megaloblastic anemia uh, or this vitamin B12 deficiency anemia, that we come to know. Okay, now the increase in mean cell volume is usually observed in case of the various alcoholic liver disease, hypothyroidism, some of the medications which are prone to do it, valproic acid, zidovudin, stavudin, and antimetabolites, and reduced mean cell volume that may usually happen due to the iron deficiency anemia, lead poisoning, and thalassemia. Now, this particular table which help us to identify the type of anemia, whether it is microcytic anemia, normocytic anemia, or macrocytic anemia. The question comes to here that why do we call it normocytic anemia? Because here what we observe that due to the acute blood loss, we may see this kind of anemia, and if it is an onset of iron deficiency anemia, we come to know whether that it is normocytic anemia. Another thing is microcytic anemia. If it is there, we need to treat it by iron supplementation. And if it is macrocytic anemia, we need to give supplementation of vitamin B12 or folate. Sometimes the alcohol abuse is also linked with it. WBC, so if it is an normal range is 3200 to 11,300 cells per cubic millimeter of blood. So if the WBC is increased, that is known as it is known as the leukocytosis, and if it is decreased, it is known as the leukopenia. If it is known as the leukopenia. Now, the leukocytosis, the increase in WBC, we frequently observed in case of the infection. Wherever there is an infection, this level is increased up to 15,000 to 18,000 cells per cubic millimeter of blood. Okay, so leukocytosis. Leukemia, here we see the greater rise. Sometimes we may see the 50,000 to 75,000 cells per cubic millimeter, but most of these cells are immature in nature. Okay, trauma, thyroid storm, that is elevated level of the T3 and T4, and prolonged corticosteroid use. Why do we observe? Because corticosteroid is linked with the immunosuppression, and it turn into the reduction into the WBC count. That's why we see this rebound increase in the WBC. Okay, it cause decrease, but once we stop it, then it turn into the release of that turn into the increase in the WBC. Many a times we see that certain conditions like emotion, stress, and seizures may increase the WBC. Um, uh, but when the WBC count is greater than 50,000, we see the false elevation of hemoglobin and MCH. As I mentioned earlier, it may link with the leukemia. Okay, it may link with the leukemia. The reduced is leukopenia. It is that is usually observed in viral conditions, aplastic anemia, and bone marrow depression due to chemotherapy or anticonvulsions. Aplastic anemia, that is again a kind of the bone marrow aplasia, bone marrow destruction, which cause this kind of reduction. That means the stem cells, or we can say bone marrow cells, or progenitor cells, weren't able to produce these white blood cells. Okay, so now the next comes into a differential WBC. As you know that the WBCs are having 5 different types: the neutrophils, basophils, eosinophils, monocytes, and lymphocytes. So they are classified into agranulocytes and granulocytes. So let us start with the first part, that is neutrophils. It may present in the blood in 2 forms, that is the polys and sacs. Now the polys and sacs, we can say sacs and bands, they are represented the level of neutrophil. If I say they are present in the form of sacs, then that is segmented neutrophils. They are mature neutrophils, and bands, they are immature neutrophils. Okay, so if sacs are there, they should be present in 36 to 73, and if they are bands, they must be present in 3 to 5%. If we see the elevation of bands, that indicates the release of immature neutrophils into the blood circulations, and that may suspect leukemia. And these neutrophils that may be involved in the pathogenesis of inflammatory disorders such as the rheumatoid arthritis and IBD. So if I say they are involved in the pathophysiology of some autoimmune disease conditions. As I mentioned, the bands are immature neutrophils. So there is an increase in the band refers to shift to the left or left shift that may occur during infection or leukemia. Many a times we are mainly rely on absolutely neutrophil count, that is known as the ANC, that indicates both sacs and bands. So if there is a reduction into the absolute neutrophil count, there is a risk of infections, because they are the soldiers, they are the frontline defense player of our body. So if their level is reduced, person is more prone to have infections. The neutrophilia, that is increase in neutrophil, that is again associated with the infections. Whatever elevation that we observe during the infection, it is something related with the conditions. Okay, the particular condition that may increase the synthesis of neutrophils, and that's why we see this kind of elevation. uh, Diabetic ketoacidosis, uremia, that is abnormal elevation of the blood urea nitrogen, emotional disturbances, burns, acute inflammations, and use of the medication, corticosteroids. The neutropenia, this is something very serious condition, and it happens with numerous immunosuppressant chemotherapy agents, uh, septicemia, or overwhelming infections. Lymphocytes, they are the B lymphocytes and T lymphocytes, so they are again immune players. So increased lymphocytes, we can say lymphocytosis associated with various disease conditions like hepatitis. Mononucleosis is a kind of the inflammation, systematic inflammation, chickenpox, herpes simplex, zoster, and viral infections. Bacterial infections like spirochetes, we can say syphilis and the brucellosis. Again, the brucellosis is the generalized infection, especially affect the reproductive organs. Leukemia, the multiple myeloma, that is the tumor of plasma, which are greatly associated with the lymphocytosis. And the reduced one, that is, it is known as the lymphopenia, significantly associated with the burns, trauma, lupus, HIV, and lymphoma. This is monocytes. They are, if they are present in the bloodstream, they are known as monocytes. Once they leave the bloodstream, they are macrophages. So monocytosis is observed in tuberculosis, subacute bacterial endocarditis, syphilis, leukemia, and lymphoma. And the monocytopenia, so we can say the most of these agents are observed in bone marrow suppressive agents and severe stress. Eosinophilia, it is normally not present in our body, but if it is present in very minute amount, hardly 4%, not more than that. And again, they are involved in the allergic reactions. So they are involved in allergic reactions, especially uh, the immune reactions related to the parasites. And this eosinophil count observed during day, in the morning and the evening. So we see the diurnal variation in asthmatic patients. Many a times we see this elevation, and this elevation, um, uh, yes, this elevation we observed in various situation, diurnal variation. So at what time you are collecting the blood is also makes the difference. Okay. Eosinophilia is usually associated with the allergic reactions, hypersensitivity reactions, vascular disease like Raynaud's disease conditions, parasitic infections, immunodeficiency, and some of the malignancy. The reduced one is associated with adrenal steroid productions. Adrenal steroid productions. Basophils, the last one, it is usually contained with the histamine and leukotrienes, and it gets elevated in hypersensitivity reactions such as penicillins, hypersensitivity reaction, or food allergy, or medication allergy. In case of the RBC cancer, it gets elevated. So these are the 5 different types of WBC, and we can use this differential WBC for the identification of an individual conditions. As I mentioned earlier, always we need to look with the associated signs and symptoms. Say, for example, if there is an allergic asthma, and if we see the basophil count is eosinophil count is increased, we may presume that it is allergic asthma. Okay, if there is a parasitic infections, there is an elevation of the basophil. These are rare of the rare markers which are used for this disease condition. Platelets, many of you are knowing very well, especially the dengue fever that caused the devastating effect on the platelets. And we see this range of 1.5 lakhs to 4.5 lakhs per cubic liter of blood. And it's an important parameter for hemostasis, that is blood clot formation. And if anything that goes below 50,000, it is alarming situation. It is an alarming situation. The patient needs to keep in a very safe position so that patient won't have any cut or any accidents or trauma. Otherwise it would turn into the uncontrolled bleeding. I see the patient where this the reduced platelet count કે સિમ્પલ આપણે જોયું છે કે આપણને બ્લેડ વાગી હોય તો આપણું બ્લડ છે એ મેક્સિમમ 5 મિનિટની અંદર બંધ થઈ જાય. I see the patient where this platelet count is less than 50,000 and blood continues to flow for more than 20 minutes. Could you believe it? So this is the importance of platelet in our body, and hence in case of the dengue we are much more we are much more sensitive to this platelet value, because the reduction in the platelet value that would turn into the intravascular hemorrhage, and patient could die due to it also. So the elevation of the platelet is known as the thrombocytosis, thrombocythemia. Some of the malignancy is also observed. We are not see this very often, elevation of the platelet, but reduction in the platelet, it is observed into the thrombocytopenia, such as in case of the autoimmune disease, we can say thrombocytopenic purpura. Why do we call it as purpura? Because we see the purple color patch on the skin. Okay, patient no platelet level ghate, so you can identify very superficially, grossly, that patient na skin ઉપર તમે જોશો purple color na patch આવે. Okay, so that is an indication, indirect indication, that platelet level is going to decrease or it already gets decreased. In aplastic anemia, or in case of the radiation chemotherapy, in such conditions we see these kind of things. Another parameter is monitoring of the coagulation. There are 3 levels which we are measured: the PT, APT, and fibrinogen. The prothrombin level, activated partial thromboplastin level, and fibrinogen level. So see the PT, usually we need to have saturated plasma. We add calcium and thromboplastin to it, and we see the time which is required to form this fibrin strand in seconds. So this PT is elevated in liver disease. So even though there is a liver disease, you see the uncontrolled bleeding to the patients, because the coagulation factors are not formed, and that turns into the prothrombin time elevation. It is also used to monitor the warfarin therapy. Okay, so one of the thing where we see the useful of laboratory data to monitor the therapy. Okay, laboratory data are used for many not only used for the diagnosis. Okay, they are also observed for the drug treatment, monitoring of the drug treatment. It is also observed for the to see the change in the physiological conditions also. Okay, so overall ongoing disease process can also measured through this parameter. This is international normalized ratio. As I said earlier, that the prothrombin time is a ratio of patients prothrombin time to the laboratory control prothrombin time. And if we need to achieve this INR for arterial fibrillation, so if it is 2.5, if it is 3 higher than if it is 3.5, it is required for the deep vein thrombosis and pulmonary embolism. It's a kind of the vascular disease conditions. And for the monitoring of the anticoagulant therapy, we need to see the clotting factors like 2, 5, 7, and 10, which are produced by the liver. So it can also use to assess the liver function. APTT, that is a two pathway, intrinsic pathway and common pathway, which is monitored through APTT, and it helps to detect the deficiency of factor 8, 9, and 11. So it is used to monitor the unfractionated heparin therapy. So it should be targeted between 1.5 to 2.5 times than the normal one. Again, I say this normal control is set in individual lab. It is not a generalized one. Okay, so if we see this, we can have the better idea. Okay, so this is all about for the complete blood count, and this complete blood count or this kind of parameters are routinely measured. Urine analysis is very useful tool for the identification of the liver disease as well as the non-renal disease. These two are the most commonly encountered laboratory investigation for individual. Okay, and based on this urine analysis, we come to know different disease conditions, whether it is related with the kidney or any other system. So urine analysis mainly consists of the gross appearance, pH, specific gravity, protein, glucose, ketones, blood bilirubin, leukocyte esterase, and nitrites. Leukocyte esterase is indicating the presence of WBC into the urine, bilirubin that identify the jaundice-like condition, blood that indicates the kidney stone-like condition or hematuria or any obstruction into the or any inflammation into the urinary tract that may shows the blood presence of blood into the urine. Ketone, that is for the diabetic ketoacidosis. Again, glucose for the to indicate the severity of the diabetes. Protein, it may indicate the improper glomerular filtration, so that protein gets filtered into the urine. Specific gravity, so again it indicates the different disease conditions. Appearance, as I mentioned earlier, it is usually a pale yellow because of the urobilin in yellow color pigment into the urine. But if it color gets different, then we may suspect different disease condition. If it is red to brown color, we may see the urinary obstructions. Okay. If it is somewhere blue, it may indicate the severe urinary tract infections. So there are several it requires the expertise also. As a pharmacist, we are not that expert. We do not have that expertise to identify the disease condition. But yeah, at least we should know uh, which condition is depicted by the urine. Okay, so pH, that is alkaline urine or acidic urine, that we come to know. Then electrolytes, just as the time we are running short of time, hardly 10 minutes are left. Just we have some outline of electrolytes and blood chemistry. The electrolytes mainly they are serum sodium, potassium, chloride, calcium, phosphate, magnesium. All of them are measured for one or another disease conditions. So there is a panel which is being identified in diagnosis world, that is known as the BMP, CMP, SMA6, SMA12, and CAM profile 20. So BMP is the basic metabolic profile, measure the sodium, potassium chloride, carbon dioxide, glucose, blood urea, nitrogen, and creatinine. The CMP, in addition to the BMP panel, we are going to measure albumin, alkaline phosphatase, ALT, AST, total bilirubin. AST, ALT पहला SGOT, SGPT ना नामे usually lab वाला denote करता था, but now they are having different name together. They are ALT and AST. They are transaminase, mainly indicating for the liver dysfunction. So we will quickly go through. We have just 5 minutes. The sodium, which is hypernatremia and hyponatremia, so it indicates the various conditions like gastroenteritis and diabetes and insipidus. Whereas hyponatremia reduction is rarely happen, but it is still observed in chronic renal failure and nephrotic syndrome. Potassium is very much important because it is mainly located intracellularly, and elevated potassium level is associated with numerous disease condition, especially the renal failure and heart. Okay, so mainly the reduction, the potassium level is also measured to monitor the digoxin therapy, uh, diuretic therapy, uh, antihypertensive drugs like ARBs, angiotensin receptor blockers, and S therapy, because they turn into the hyperkalemia. So we need to see this hyperkalemic incidences when the patient is taking these medicines, and patient and doctors are always advised to have this potassium level when patient is on ACE inhibitors or ARBs. We need to be very cautious when we measure the potassium. We should not measure potassium in hemolyzed blood sample. Uh, Potassium level is also decreased in case of the severe dehydrations like diarrhea, vomiting, Cushing syndrome, respiratory alkalosis, and amphotericin B, that is related with the nephrotoxicity. Uh, it is chloride level, so it is having important role in acid base balance and fluid balance. So it is used to identify the acid base balance disorder. So hypercholemia and hypocholemia that we need to investigate for certain drugs, especially the anti-ulcer drugs. Glucose, we are very much aware with this diabetes. So if the fasting blood glucose level is more than 126 and random blood glucose level is greater than 200, then it is diabetes. Uh, that is 100 to 125 and more than 140 random blood glucose, that indicates the impaired glucose taste, or we can say pre-diabetic patients. Hypoglycemia is observed because of the fasting conditions, oral hypoglycemic agents, or insulin over. Uh, Blood urea nitrogen, it is an indicator of the chronic renal failure or any kind of the nephrotoxic medicines. If we are taken, then it shows this kind of level. Same as creatinine, which is also associated with the renal dysfunctions. And we see this because of the muscle damage, it gets also elevated. In case of the myasthenia gravis, it's an autoimmune disease condition, it gets elevated. Reduced creatinine, where we see the muscle spinal cord injury, it is also decreased. In case of the cachexia, is the muscle wasting in the patients with the cancer. And in elderly patients, we see the reduced creatine because muscle mass gets reduced, protein intake is reduced. So we couldn't see the creatinine in the blood, okay, because protein won't metabolize, so that creatinine level is not elevated. uh, as the time is approaching, I see this is the last slide related with the creatinine. We need to investigate the creatinine clearance, because we need to adjust the dose in the pediatric patients. And if the patient is taking if patient is having renal failure, for the dose adjustment, we need to take this equation into the consideration. So very much thank you very much for patience listening, as this is the last lecture. You are almost having 5 sessions in a day, so you may have almost exhausted informative day for this refresher course. So there are various tests I still require to discuss, but due to this time constraint, I couldn't be able to include all these parameters. Even though it is very much difficult to cover all this parameter, even though just I am telling you the importance, it's very monotonous way. But yeah, if you are having any question related to any laboratory parameter which are not included in this lecture, you can ask me. Yes, is there any question? Thank you very much for your patience listening.
Thank you, sir, for the very informative session. Sir, we are having few questions for our participants. First is, if the patient is taking chronic medication, does it affect the laboratory data or parameter?
It depends on which medication patient is taking. Okay, if I say the patient is, I'm taking one of the example of chronic therapy for the hypertension. If patient is taking thiazide diuretics and ACE inhibitors, by the physician's experience, the potassium level is going to alter. The thiazide diuretic cause hypokalemia, and the ACE inhibitors are causing hyperkalemia. So all in all, the potassium level is normalized. Okay, so yes, potassium level is a fact on a longer run, Uh, but in most of the instances, we need to check the particular laboratory parameter for a longer run. Say, for example, statins. Statin affect the creatinine kinase level. Okay, so if we want to monitor the statin therapy, muscle wasting is unusual adverse effects of the statin. So we need to monitor this creatinine kinase for a longer period of time if patient is on statins. Yeah, it is affecting. Yeah.
Okay. Next is that they would like to know about some information with respect to oral insulin and its current status.
Oral insulin is not available because the insulin is degraded into the GIT, and there are many research papers which are published in recent years, but I really doubt the commercialization of this oral insulin. In preclinical study or some preclinical study, they may show the effective result, but I really doubt its clinical effectiveness and the scale up procedures.
Okay. One more is there, sir. Which time the maximum amount of enzyme release and why? Or it indicates the benefit or harm to the body.
uh, Enzyme level is a very if we talk about the normal homeostasis, the enzyme gets released, and that's why we have the normal value. Say for example, AST and ALT, its normal value is 0 to 40 units per liter of blood. Fine. So it is quite obvious that enzyme gets released in our body, and that because of the routine procedure, I would say, the liver cells or hepatocytes that undergo the process of aging, and that may undergo the process of apoptosis, and that release the enzymes, and that's why we see the enzyme, but it usually won't harm the body. It's related to the normal homeostasis. But if any insult which happened to the organ, the enzyme, there is a rush of enzyme which we observed after 6 to 8 hours. Usually we need to provide 6 to 8 hours for the effective for the effective measurement. Yeah.
Okay. One last is there, sir. What is the role of CPK test and D dimer in COVID-19 diagnosis?
Basically, D-dimer is the test which indicates the thrombosis process. As COVID-19 is considered to be a thrombovascular disease condition, and patient may end up with the intravascular coagulation, and that may produce the devastating result and many deaths due to that. So in order to monitor this coagulation process, the D-dimer is measured. D-dimer is the end product of fibrin, okay, fibrin degradation. So if there is an elevated fibrin level, the D-dimer level is elevated. Yeah.
Okay. A few questions are also there in the chat. Why antibiotic azithromycin is and the antiviral both are prescribed in COVID treatment presently?
Both of them are having different target. Okay, I'm really sorry why azithromycin is implied, but I need to see the or I refer one of the biochemical pathway where the azithromycin is really effective to reduce the viral load. And the antiviral, especially if I say the favipiravir or the any other antiretroviral therapy, they are there for inhibition of the RNA dependent RNA polymerase, so that the viral won't produce viruses won't produce. Yeah, ivermectin, the role is still in a questionable. Some of the reports suggested that it prevents the entry. Remdesivir, again, it prevents the assembly of the virus particles, so it won't get released into the bloodstream. So the assembly of the virus particles that get affected. Anticoagulant, as I mentioned earlier, the D dimers related to it. So the D dimer is indicator of the coagulation. So if you are given anticoagulant, that may prevent the intravascular coagulation. Very good question. What about dexamethasone? yes, dexamethasone is really effective if your CRP level is significantly elevated. I would say that dexamethasone is one of the life-saving agent in COVID-19, in advanced COVID-19 patients, because there is a phenomena called cytokine storm in the COVID-19 patients, and that's cytokine storm which is inhibited by the dexamethasone. So various effective role of dexamethasone in COVID-19 is very important drug. That the one of the report the previously it was said by the WHO that the dexamethasone should not given to the patient, but in various studies in Italy, where we see the significant mortality somewhere in March and April, and the Italian physicians were tried this dexamethasone in COVID-19 patients, and they found beautiful results in those patients, and mortality rate is significantly reduced in those patients. So that dexamethasone is great drug. um, Doxycyclines are having some off-label use related with the immune modulations. So I'm not that expert that why doxycycline is used, but yeah, immunomodulating role of doxycycline may have this kind of, yes. Prednisolone instead of dexamethasone, yes, we can. Both are, dexamethasone is much more potent form. Prednisolone is less potent. Dexamethasone is given by intravenous route and very fast onset of action, I would say. So yeah, dexamethasone can be given. Yes.
Okay. So,
Uh, okay. Yeah.
okay. Okay. Sir, thank you very much once again for inviting our invitation as a resource person, and thank you so much for the informative session. And we have reached
Thank you
towards the
very
end.
much for IICP, and thank you very much to Harsha ma'am, management of CVM, Usman sir, for inviting me to deliberate the session. Thank you very much.
Pleasure
Thank you
is
so
all
much.
ours, sir. Thank you so much. Uh, we have reached towards the end of the first day sessions. I hope you all the participants have enjoyed the very informative sessions today. Thank you all. Have a great day ahead, and see you all tomorrow by 9:30 in the morning. And kindly join through the Zoom meeting before 15 minutes, like 9:15 to 9:30 a.m. in the morning. And link will be shared to you guys very soon. Thank you. Thank you. Thank you one and all. Bye, man. Bye, sir.